Rachel McCrea is a professor of statistics. I'm reading this from my notes, by the way, so apologies that I keep looking down. Rachel is a professor of statistics at the University of Kent in Canterbury and is the head of the statistics group. Her research is focused on the analysis of capture-recapture in ecology. She has a book on it. In particular, Rachel has been awarded many awards and positions. So, for example, she completed a fellowship in 2016, funded by the Natural Environment Research Council, also commonly known as the NERC, and has been awarded an EPSRC New Investigator Award, I believe, in 2018. She's also received the Royal Statistical Society 2020 Guy Medal in bronze, and has recently been appointed as a Fellow of the Learned Society in Wales in 2021. So, Rachel, if you would like to take over now, please.
Thanks very much, James. And yeah, thanks so much for the invitation to talk today. So it's really interesting coming to sort of give a presentation in an event like this, because in many ways I still very much consider myself an early career statistician, although I'll have to do the calculations to see if that's within 15 years of graduation. So I'll just share some slides. Okay, so hopefully that shows up. Please do pipe up if it doesn't. So what I thought I'd talk about today is really about how I've kind of navigated my career path. I think it often feels, when I sort of look back at the last 10 years, that a lot of this has been down to luck. So actually putting together this presentation really made me think about, well, what gave me those opportunities? And hopefully some of this might be useful when others are thinking about what types of grants or what types of collaborations to think about. So I very much think that my career is still evolving. So this is where it's got so far, and I'm not sure what I'll be doing in 10 years' time. So within the presentation today, I'm just going to tell you a little bit about me, because I think it's really important to know the person behind the career. I'll tell you a little bit about what pathway my career has taken to date, so what positions I've had. And what you'll see is that collaboration is a really fundamentally important part of the research that I do. So I'll talk about how I've developed those collaborations. And then to put into context how those have sort of contributed to my research, I'll talk a little bit about some previous research I've done, some of the current projects I'm working on, and what I really want to look at in the future. So, as James said, I'm Professor of Statistics. A lot of my research has been focused on ecological statistics. So ecological statistics was something that I actually encountered during my Masters in Statistics, when Byron Morgan, who is a sort of eminent professor at the University of Kent, gave a module, and I kind of refer to it as a fun module about ecological statistics. And I often tell Byron, because he went on to supervise my PhD and has been a really good mentor since then, it was my absolutely favourite module, but I didn't answer any of the exam questions on it because it was just too challenging. And from there, starting with the idea of capture-recapture, and I'll talk a little bit about that when I talk about my research. More recently, I've got involved with some statistical modelling for the social sciences, and that's all about trying to estimate hidden human populations. As James has said, I took over as head of the statistics group at the University of Kent in 2020. So that was wonderful timing with the pandemic. And that's kind of been a new sort of leadership direction for me. So that's very much been a kind of step change in my career. So I'm 39 years old. I think once I turn 40 later this year, maybe I won't put my age on these slides. But I also want to say that, you know, I've navigated my career while being a mum. Okay, so I'm very open about the fact I've got 3 children. I've got 9-year-old twin girls and a 5-year-old son, and we've got a menagerie of animals, a dog, guinea pigs, and lots of fish. So I have a really busy home life as well as a busy work life. So what sort of pathway have I taken to date? So I mentioned that I did a master's in statistics. So my undergraduate degree was in mathematics in Cambridge University and St John's College. And then I had to think about what I wanted to do. And to be honest, I was a bit confused about what I wanted to do. But I'd quite liked statistics, and I'd had an absolutely amazing supervisor in St John's College, Rich Samworth, who's now a fellow of the Royal Society, so he's a really eminent statistician. And he said, oh, you know, go and do a master's in statistics. So that's what I did. So I went to the University of Kent, and basically I never left. So I did my master's 2003 to 2004, and then stayed for a PhD in statistics. And the PhD was joint with the CNRS in Montpellier. So that was the first sort of collaboration I had, building links between the University of Kent and another organisation. Then I had 2 postdoc periods. So in 2008 to 2010. Now, at that stage, it was actually really uncommon to stay in the same institution. And I think it was also one of these things that people didn't really like to see. And I know that this is even more challenging in mathematics, but it did come down to kind of a practical situation that my husband worked at the University of Kent. And frankly, we didn't want to move at that stage. So in a way, I think I probably had extra time as a postdoc, because it would have been possible for me to move elsewhere to be a lecturer, but it was really about finding opportunities that would work for me at the University of Kent. So I then got a NERC postdoctoral fellowship, and I'll talk a little bit more about that in a moment. And then actually during that fellowship, some lectureships became available at Kent, which I applied for, and I postponed the start of that until the end of my NERC fellowship. And then I became senior lecturer in 2018 to 2020. And you'll see that I then jumped straight to professor in 2020. So that was again a very big jump for me. And that was in recognition of my Royal Statistical Society award. So what I wanted to point out was what have been the kind of key drivers in this career step change. So the first thing I want to talk about is this red box. So after doing my PhD in statistics and getting an internal postdoctoral position, I was very aware that if I wanted to stay in Kent, I needed to kind of get money to allow myself to stay in Kent. So I applied for an EPSRC fellowship, and it did really badly. I mean, really badly. So I got really low scores, and was then sort of questioning, am I cut out for academia? And I'll come back to this point a bit later. So obviously it's always really upsetting when you fail at something, but I kind of had to pick myself up from that and think, well, if I do want this to work, I need to sort of look for other opportunities. And this is when I started thinking, well, actually, is EPSRC the only option for me? Because some of the reviewer comments had sort of commented on, is this really, you know, statistical research? Is it really mathematical research? Or is it just sort of interesting new applications? So that's when I had to look at NERC opportunities, and I applied for a NERC fellowship. And the key part about all of the grant applications I've applied for and been successful with since then, project partners have been a really sort of strong feature in them. So because I wanted to stay at the University of Kent, I was really having to demonstrate why they should fund this fellowship, because it would allow these new partnerships. So for example, for my NERC fellowship, I partnered with Imperial and the Alpine Wildlife Research Centre. And with my later EPSR Senior Investigator Award, I partnered with 3 conservation groups. And then more recently, I've got a Leverhulme Fellowship, and I've partnered with New York University and with Traffic, which is an organisation that collects data on wildlife trade. Okay, so as I've said, my research is really motivated by real world problems. And the reason for that is that I'm interested in developing new statistical methodology which can influence decision making. So I really fundamentally believe that decision making should be evidence-based, and I'm sure that's a sort of view in common with a lot of people listening today. But because of that, there's no point me just developing new statistical methodology in isolation. I really need to be working with the people that are going to be able to make that step change within decision-making processes. So putting together this presentation, it made me evaluate, well, how have I developed those collaborations? And really, I think I've sort of identified 3 main features. The first is that all of my colleagues will tell you, I love going for coffee. And some of those kind of informal chats over coffee have resulted in some of the best research projects. So those might be coffee chats with people within the statistics group at Kent, they might be with people from other departments at the University of Kent, or they might be visiting seminar speakers and so on. So just being able to talk to people about their research ideas and their what they happen to be interested in and what their unanswered questions are, those are really sort of valuable times. Early on in my career, it was also especially important to attend conferences and put myself out of my comfort zone and present my work. Obviously, that was something that I found really daunting to begin with. But now I realise actually those sort of questions that you kind of dread at the end sometimes result in the most interesting research, because they're giving you a sort of perspective that perhaps you didn't have. And then the third one is when I started to run workshops for people to learn about statistical methodology. So workshops have kind of been a feature of my academic career to date, really since I was a PhD student. So as James mentioned, I've written a book on Capture-recapture, and when we were writing that book, we thought, well, actually, we should put together some workshops and test this material on people. And it's meeting people through those workshops that have resulted in some of the collaboration research projects that I'm going to talk about today. So I think if you're interested in sort of very much pursuing an interdisciplinary research career, what you've got to realise is that your statistical or mathematical expertise is so valuable to researchers in other fields. There are, however, sort of some difficulties, because quite often when you work with people in other fields, they'll very much use different terminology. And I actually find this sometimes just speaking with James and Edward, who are on the ECM committee, that they sometimes have sort of more mathematical terminology and I would have the more statistical terminology. But actually being able to develop the ability to talk about your research in a non-technical way means that you can kind of bridge that language barrier that disciplines might have. The other really exciting approach that I've taken is to sort of look out for co-supervisory opportunities of research students. So although I've only got, I'm not sure how many students who I've sort of primarily supervised, I've also kind of informally co-supervised a lot of students from conservation ecology groups or from social science groups. And that's been really, really interesting, a really useful way of getting to know new research areas. And then, as I mentioned at the start, there is very much at the moment a challenge of trying to balance the conflicting demands of academia, you know, whether it's getting the publications that you need to apply for your next job or apply for promotion, whether it's the teaching commitments and dealing with going to remote teaching and remotely trying to support students. You know, all of this is using our academic time. So we have to be able to think about, you know, how am I going to balance those different parts of my career? So that's a little bit about me and about how my sort of research has taken the path it has. So I'm going to talk a little bit about some of the research that I've actually done. So I'm going to start with talking about some previous research. So it starts with some of my really early work on Capture-recapture, and then 2 or 3 projects that have sort of stemmed from that. Okay, so Capture-recapture data, apologies if those of you in the audience are familiar with it. But basically, capture-recapture of ecological populations relies on the animals that you're surveying being individually identifiable. So that might involve attaching some kind of mark to the animal. So you'll see on this cormorant, it's got a ring on its leg, and that ring has an alphanumeric code which individually identifies that bird. Or it might be on this straw-coloured fruit bat that they've got this radio transmitter collar, so that when the recording device gets close to it, it will tell you which animal that is. So those are kind of examples of artificial marks, but there are some species which are naturally identifiable. So adders, for example, there's lots of studies of adders in the southeast where we are, and they are individually identifiable by their pattern. And this is a great crested newt, and you can see this sort of striking yellow and black belly pattern. That black and yellow pattern is like a fingerprint for that newt, so it individually identifies that newt. So the type of data we record is basically vectors of binary information. So for each individual in this, which we've encountered in the study, we'll have an entry of 1 or 0 to say whether that individual has been seen on that encounter occasion. So for example, this top individual here has been seen on occasions 1, 3 and 4, but wasn't captured on occasion 2 and 5. Now, sometimes you can also collect additional auxiliary information. So for example, if your study area is split into multiple discrete areas, you might record which area an individual was encountered in, and we'd denote that by a letter instead of just binary information. Alternatively, it might actually reflect the state that the animal is in when it's being captured, so say breeding or non-breeding state. Now, the issue with capture-recapture data is that we've got imperfect detection. So you can see here that we've got some zeros, but this individual must have been alive at time 2. We just failed to capture it. So we need to take into account that imperfect detection to be able to estimate the parameters that we're actually interested in. So our detection probability is seen as a bit of a kind of nuisance parameter, but it's essential to incorporate that into the model for us to be able to estimate robustly survivals, population sizes, or transition probabilities between states. Now, I've sort of over the years developed a number of new models for capture-recapture data, but one particular area where I've contributed is in the multi-state capture-recapture modeling framework. So I've, in collaboration with others, so this is not just all me, we've developed new models both for closed populations where we assume that there's no birth or deaths or immigration or emigration, and also open populations which allow for new arrivals and departures. And the types of applications to these models are sort of huge, but 2 just to mention. So we've looked at the recruitment of grey seals into breeding populations off the Scottish coast. And we've also looked at disease progression in a chytrid outbreak in the mountain chicken frog in South America. And the reason I've put this up, I know there's a number of you who work in epidemiology, and fundamentally the types of models that multi-state capture-recapture models are very similar to models for epidemiological data. So at some stage I'm really interested to explore those parallels and understand it a bit better. What I want to mention here, you'll see that on the slides there's kind of different organisations that I've been involved with, and this mountain chicken frog collaboration actually came about. So one of the capture-recapture workshops that I ran, somebody who was attending that got chatting to me afterwards, was talking about their data, and then invited me to sort of be involved with that project. And that's how my first link with the Institute of Zoology started. So being able to put yourself out there and kind of provide a service and run workshops, it really does open up opportunities. So multi-state capture-recapture models can become very complex very quickly. So this is just a diagram to represent a cormorant capture-recapture study that I looked at. Is actually during my PhD, so many years ago now. But in that study, there were 3 different colonies, and there are also 2 states of birds, so breeding and non-breeding. And as well as capturing live individuals, they also recovered dead individuals from outside of the study areas. So the actual sort of number of transition probabilities we're trying to estimate, which represented different types of ecological process, became complex very quickly. So because of that, it's actually very difficult to estimate all those transition probabilities from capture-recapture data alone. So what we looked at was something called multi-state integrated population models, which basically unifies the analysis of capture-recapture type data with the analysis of time series type data, so count data. And the idea behind this is that count data essentially contain information on survival and transition probabilities. And so by simultaneously analysing those 2 types of data set, you can better estimate the survival and the transitions. Now, these models become complex very quickly, so we've done some investigation on whether these models are parameter redundant and what data is needed to prevent those models from being parameter redundant. And the concept behind an integrated population model, you're taking a likelihood function, so which is a probability, and another likelihood function, which is a probability, and we're needing to multiply them together. So in order to be able to multiply 2 probabilities together, clearly that means we're assuming independence. So I've done quite a lot of work, and that's what my NERC fellowship was on, looking at the validity of that dependence or independence assumption and what actually happens if your data are dependent. Okay, so that's where I kind of started with capture-recapture data. And within the University of Kent Statistics Group, we run something called the Stat Desk, which is a kind of consultancy service for other academics within the university. And it was one of those discussions that somebody came to me from the conservation ecology group, DICE, that I'd already done some work with. And they came to talk to me about removal data. Now, removal data arise, or they originally arose because basically people wanting to develop building plots and put up new buildings needed to ensure that any protected species that were on those grounds were safely removed before the building commenced. So quite often great crested newts kind of hit the press because they prevent developments from happening, but they don't actually stop developments from happening. It just actually delays things a little bit. So with removal data, what you have are, on a number of occasions, ecologists will go out and they'll go and try and remove as many newts as they can find on a series of consecutive days. So the data themselves are really sort of simple count data. So we've just got a count NT over time. And intuitively, what you'd expect is that you start by capturing a lot of individuals because there are going to be lots of individuals available. But as you've removed more and more, you're going to encounter fewer. So what you see is this kind of decline over time, but you'll see here that it's quite bumpy. So if you think about this as the population is completely closed, so there's no new arrivals, no departures, and if you assume that the probability of capturing each of the individuals is the same, what you end up with is a geometric model. So that's this blue line here. So that's the geometric model fitted to the removal data of the great crested newt there. And you can actually adapt this a little bit further to improve it. You can say, okay, well, maybe the capture probability is influenced by the weather conditions, for example. And by doing that, you can say that we regress, have a logistic regression of the detection probability at time t is related to some weather covariate information that you've collected. So in this case, it's minimum air temperature that's been recorded on that day. And you can see that the green model here that we fitted to the data is a much better fit than the blue model. But that's all sort of very, very nice, but it still doesn't really address problems like this peak. So you can see it around 39, we've suddenly got a much larger number of newts that we've captured. So we need to think about what's going on there. And the person who had come to see me at the Stat Desk consultancy came with this data. So this is some removal data of common lizards, and you can see that this looks nothing like a geometric shape. And the person came to me and said, oh, well, I fitted the geometric model to this data, but I'm not sure it's the right model. And sort of you, as an applied statistician, when you're sort of working with these people, you sort of say, oh, well, I'm very glad you didn't just go with the results from fitting a geometric model, because that's clearly not right. So what's actually going on here? And what we had was a conversation sort of thinking and discussing about what might be wrong with the geometric model. And it turns out that these populations are basically unlikely to be closed. So what happens is that these removal studies are basically started as soon as developers contract the consultant to remove the individuals. And so what happens is that the removal study actually takes place during the breeding season of the lizards. So this giant peak here actually represents the juvenile individuals that have been born into the population that you've then removed. So how did we go about modelling this? So what we did, we said, well, suppose that we've got individuals arriving in what we call renewal groups, so groups of arrivals, and we're going to model the arrival pattern of those groups using a normal distribution. And I'm not sure if we've got any statisticians in the audience, but just in case we have, basically, if we don't know the number of renewal groups that we might need, we can build in a reversible jump Markov chain Monte Carlo algorithm to select the best model, essentially. So you can move between models with different numbers of renewal groups. And it worked really nicely for that. And that was great. And we sort of published that work. And then we had another coffee chat, and they said, oh, yes, well, it's really nice that we've done this, but you know, the lizards actually do something else as well. And what they said, well, the lizards might go underground. So essentially what might happen is that you've got what we refer to as an underground city. So you've got the lizards, which are on top of the ground, which you're actually in your kind of detectable part of the population. And you've got those that have kind of decided to go underground. And to approach sort of modeling this, basically we developed a two-state model where one of those states is unobservable. So the underground state means they can't be detected. And this sort of naturally fits into something called a hidden Markov model framework. But the difficulty here, I touched on the concept of parameter redundancy and which parameters can we estimate and how much data do we need to be able to estimate them. If you just try fitting a two-state model with one unobservable state and you've just got individual counts, you'll actually hit parameter redundancy. You won't be able to estimate all of the parameters of interest. So what you need to do instead is have what we refer to as a robust design removal model, where you have primary samples, primary occasions really, where you can assume that the population's open between those samples, but then you have secondary samples, and between the secondary samples you assume population closure. So for the case of the lizards, they actually had multiple visits in a day, so we assumed that the primary samples were the days and the secondary samples were each of the visits during the days. Now, as well as sort of being interested in developing models for particular applications and kind of adapting models for that purpose, I'm also sort of interested in some kind of main statistical theory as well, sort of developing new tools which can be used for a wide range of statistical models. And model selection and goodness of fit assessment are two areas where I've done quite a bit of work. So the types of models I'm looking at, both the multi-state capture-recapture and also integrated population models, it's non-trivial to essentially work out what the best model for your data is. So I've developed various approaches to address that. And similar with goodness of fit assessment, that you can develop lots of new statistical models and you run the risk of them being used blindly by ecologists because they're sort of readily available in R packages and things like that. So what I've worked on quite extensively, building actually on those early collaborations that I had with CNRS built up during my PhD days, we've gone on to develop diagnostic tests which can be performed on your capture-recapture data before you actually do any model fitting to determine if the model you're about to fit is actually suitable or not. So the diagnostic tests explore for things like capture heterogeneity, whether there's evidence in the raw data that some individuals are more detectable than others, tests for different types of transition probabilities and behaviour, and also some work determining the number of states for a hidden Markov model for the special case of partially observed capture-recapture data. Okay, so I'll just keep an eye on time. I'll talk a little bit about some of the current projects. So these are very much projects which are my PhD students are generally leading, but I want to mention them because they really have kind of been exciting parts of my career, and they link in with the grants I've got at the moment as well. So the lesser night gecko is a tiny endemic reptile in the only found on the outer islands of Mauritius. And it's really challenging to detect, and it's basically becoming extinct on certain islands in that region. And the IUCN has deemed that this species is vulnerable, so there needs to be action to prevent the complete loss of this species. So the aim of this project is to re-establish a population of lesser night gecko on one of the islands surrounding Mauritius, which is Round Island. Now, some aspects of this work, so on removal and reintroduction modelling, that's the topic within my EPSRC New Investigator Award. And this project is in collaboration with Durrell and the Mauritian Wildlife Foundation. So the project partners out there are kind of doing the kind of practical aspects. My PhD student she was returning on Monday, having just spent 6 months out in Mauritius doing some field work. So that's the aim. But there's kind of quite a lot of statistical questions here. So they're going to be taking the lesser night gecko from Gunner's Coin, which is this island here, and they're going to be translocating them to Round Island. So this kind of links back to some of that removal modelling that I was talking about before. Removal modelling for the purposes of land development is very different from removal modelling of a source population, which you're hoping to then translocate elsewhere. So what we need to be sure of with the removal modelling on the source population, we want to be able to address, well, actually, we don't want to kill off the source population by taking too many of the individuals and translocating them to another island. So we need to think about the impact of that. We need to think about modelling to forecast the impact of the translocated lesser night gecko on the other existing species on Round Island. And in order to address both of these, we've got some historical data on a previous translocation that was done from Île Vracois to Île Marianne. And we've also got lots of data on different species on Île Marianne, so that you can kind of look at the impact on the other species. And then the other part that they're really interested in, once they've done the translocation, how should they go about monitoring the different species on the island afterwards? And then really, I think probably one of the most challenging questions is, how do we determine if the project has been successful or not? So what would be the project success criteria? Obviously, we want lesser night gecko to establish themselves on Round Island, but there's also other factors to consider in terms of the effect on the other species. And we've all known that we've got to be kind of resilient during COVID times, but my PhD student was kind of hit another curveball when there was a massive oil spill off the coast of Mauritius in the summer of 2020. And so a lot of her work now has been slightly adapted to look at the effect of the oil spill on the reptiles on the island. And Jersey, which is where Durrell is based, made the decision that actually some of the reptiles from the island, some of the endangered species, should actually be translocated temporarily to Jersey to ensure their health and security. So we're looking at whether that was the right decision to make and the potential knock-on effect of that. Okay, so the second current project I just want to mention is butterfly monitoring. So this is very much thinking about data that arises in spatial and temporal scales. And we're working with butterfly conservation. And my PhD student, James, who is doing this project, he's funded through a doctoral training partnership. And this is a really nice example of external organisations working closely with me, but also PhD students. So really bringing up those and developing those interdisciplinary links from an early stage. Sorry if you can hear the dog barking in the background, it's a busy Saturday here. So the UK Butterfly Monitoring Scheme is the world's biggest and longest insect monitoring scheme in the UK. And it started in 1979, and there's now almost 2,000 sites which are recorded with data once a week. So the purpose of this is James was wanting to develop a method to estimate lifespan of butterflies using those the data from the citizen science monitoring scheme. And we essentially had data that was recorded in a weekly format, so sampling was done once a week, but we're in particular interested in what if lifespan's less than a week. And so we developed methods which actually take advantage of knowing the day of the week in which the data was collected. Because although it's presented in weekly format, actually it's not always 7 days that's between the observations that were made. And then the other aspect of this project is thinking about how things vary on the spatial and temporal scale. So these are the incidents of the marbled white butterfly in 1994 compared to 2018. But we've got to take into account that actually fewer sites were sampled in 1994. So this doesn't all necessarily represent a spread. It might actually represent that fewer sites were sampled. But what butterfly conservation are interested in is whether at the moment trends of butterflies are looked at on very much a national scale, but can they be looked at a smaller spatial scale, and ideally develop a way of developing site level alerts. So if there's really a concerning trend in one particular site, whether they can have management intervention. Okay, so I'll talk very briefly about this because all of the work I've talked about so far is on ecological modelling. So I first became interested in trying to estimate in human populations. I was invited to a working group meeting up in Edinburgh in the ICMS by somebody from the Walk Free Foundation. So she was bringing together expertise of people who estimate population size, but from lots of different disciplines. So there were lots of social scientists, there were lots of statisticians, some ecologists, and it was a really interesting meeting because it brought together really different perspectives. So obviously it's of interest to estimate population size in, say, healthcare, criminal justice, and it shares that feature in common that you've got imperfect detection. So unlike ecological capture-recapture data, you're not physically going out and trying to capture and recapture humans, but you're trying to detect humans on different administrative lists. And the type of data that we end up getting, if these SI are the administrative lists 1 up to 4, you'll have individuals who are only detected on list 1, represented by the count of N1 and 000. Now, interestingly, capture-recapture methods for social science have kind of developed in parallel to ecological capture-recapture models. And so there's very much been 2 different pathways of development. So one thing that came out of the meeting in Edinburgh was a special feature of the journal Crime and Delinquency, where we really looked for opportunities to cross-fertilise those 2 research areas. Now, fundamentally, the model that's used for estimating human populations is simply a log linear model. So it's a really sort of basic type of model. And what we're really interested in estimating is what we refer to as the dark number, so the number of individuals who are not encountered on any of the administrative lists. And it's really important that thorough model selection is implemented, because you can have vastly different estimates of those hidden individuals depending on what structure model you fit. Now, one sort of current piece of work that I won't go into too much detail, but basically the types of hidden population that we're working with are very vulnerable individuals. And because of that, we don't want any specific individuals to be identifiable through the data that's disclosed to us as statisticians. And so because of that, sometimes when you've got small counts of individuals that are on certain combinations of lists, they censor that information. So some work with my colleague Ruth King in Edinburgh at the moment, we're looking at how do we deal with interval censored data. So for example, here in this data set, these stars could be the values between 1 and 4. So they're not telling us exactly how many individuals those are. Okay, so I'll just talk very briefly about what I'm thinking about next. So I touched on the fact we've got a Leverhulme fellowship, and this for me is the first time I've sort of put together a grant that's really sort of aiming towards more of that sort of slightly social science direction, but it is bringing in some ecological expertise as well. So the purpose of this fellowship is going to be developing new statistical tools to analyse elephant ivory seizure data, which is held by ETIS, which is the Elephant Trade Information System, which is looked after by Traffic. And what Traffic want is the potential for the statistical model I'll develop to be able to investigate different enforcement strategies on this illegal trade network, to be able to better utilise the money they've got to have a positive impact for the elephants. So the actual modelling idea actually stems from some of the multi-state capture-recapture models I've worked on. And at a conference I met Professor Porphyry from New York University, who does a lot of work on human migration. And so during the fellowship as well, I'll be looking at possible ways of investigating how those mathematical models for migratory behaviour could be incorporated into these types of models. And then there's potential extensions to this, that the model itself is not restricted to elephant ivory. It could be used for other types of data. And I met a lady from the UNODC who looks after the heroin seizure data, the UNODC, at that Walk Free meeting in Edinburgh. And she's particularly interested to look at the potential for drug seizure data. And then some other current projects, so just through conversations we've had, and Edward, who's on this call today, we've had 3 meetings with 3 organisations, and just those very short conversations we've had with them, there's so much potential. for the use of statisticians and mathematicians in collaborations with them. So it'll be interesting to see where those evaluation of sort of cyber-enabled wildlife trade research goes. So I just want to finish on a couple of points. I'm sorry, I haven't left a huge amount of time for questions, James. As I said at the beginning, I often sort of think back to the last 10 years and think that a lot of the aspects I've looked at are luck. And I think academia does come down to luck. We all know when we get a paper rejected and how heartbreaking it is. But we've got to kind of normalize failure and rejection. That I often have papers rejected. That rejection of the first EPSRC fellowship I went for, it really knocked me. But now if I get a sort of failure, yes, you can feel glum for a couple of days, but you really do need to pick yourself up again. And I'd really encourage ECMs to kind of engage with resilience training and things that universities do put on. And as well as that, although I'm seen as an interdisciplinary researcher, it is okay to do research that's not collaborative as well. You don't have to put all your eggs in one basket. So a lot of the kind of model selection and goodness fit work I've done, it's slightly more sort of fundamental statistics rather than interdisciplinary. It's very easy as an applied researcher to overcommit to too many projects. I'm sure there are times, especially during COVID, that we all felt I've just got too much to do. But it's better to kind of focus on a few and do them really well than to grow your portfolio too quickly. And finally, just really do prioritize projects that you're genuinely interested in. So at the end of the day, you're going to do your best research if it's something you feel really passionate about. And just a sort of note that something I've observed from universities, I think there's a lack of recognition within universities about the extra effort and time that interdisciplinary research can take. And to make sure that you have expertise of a lot of different funding opportunities. So you will have seen that I've sort of looked at EPSRC, NERC, Leverhulme. You know, it really is possible to look at a really broad spectrum of things. And finally, these are just my experiences. So I'm not saying that people should go out and follow this, but I always found these presentations quite useful to see what, how have you navigated it, and how did you make these things work? And I will finish there. Thank you very much.
Thanks a lot, Rachel. This is going to happen throughout the whole day, but I'm going to end up changing what I want to study for the rest of my life, like each talk probably. So does anyone have any questions? There's none in the chat at the moment. I have one or two, but I want to open it up to the room first to see if anyone has any.
I think Rosanna's got their hand up. Does that mean they've got a question?
Oh yeah, I didn't even look at the hands. Yeah, go ahead, Rosie.
Hi, thank you, Rachel. I loved your talk.
Thanks, Rosie.
I guess my question is about how you have carved out time to focus on developing fellowship applications, and if you could give any advice on that.
I would say it's one of the hardest things to do. And I was kind of fortunate that when I applied for my NERC fellowship, I was still a postdoc. And I had a really supportive postdoc supervisor who basically allowed me to use that postdoc time to develop my career, essentially. And he wasn't completely focused on, you've got to do this project, you've got to get this outcome, this outcome, or this outcome. More recently, my EPSRC grant, I wrote when I came back from maternity leave. So we had an amazing policy in our department, and I would endorse everybody to go out and encourage your departments to do this. We basically have a system where you have a reduced teaching load when you come back from a period of absence. So that might be sick leave, that might be maternity leave, parental leave. And having that reduced load allowed me to have that time to really think a project through. And I would say it's becoming harder and harder in academia to find that time, because you have so many daytime pressures. And I hate to think of anybody feeling the need to work in the evenings on what should be their day job. So I do sort of block out days in my diary. I don't always manage to stick to it, I have to be honest. But I'm not, given that Edward's in my group, so he might have comments on whether I disappear for a period of time. but I think it's really important for everybody to feel it's possible to not answer email for a day, to not be on Teams for a day and to actually say, no, I'm focusing on that. Because what I find it incredibly difficult to just do an hour of research here or there. I need time to kind of get into something. So yes, I think during term time when I'm teaching, it's practically non-existent. Then during sort of outside of university term time, which doesn't clash with my children's school holidays, that's the time I'm probably most productive in terms of writing proposals.
Thanks for the question. That I had that on my list, so that's ticked one off. Does anyone else have any questions?
I have a question, but I'm happy to let other people go if they want to.
Go ahead, Nick.
Not necessarily relating directly to your talk as such, but yeah, which was really, really interesting, and talking about identifying animals by their unique patterns.
Yeah.
Is particularly interesting for myself. We do through my job a lot of motion capture of various components on test, and we have to put dots all over them, make sure we can identify them, they have all their unique dots, etc. And I also keep koi fish, so having a software which could tell my koi fish apart would be very interesting, but that's by the by. That was a really nice bit to see there. Earlier on in your talk, you mentioned that a lot of your career has been by luck, and that really caught my attention because whenever I'm still an ECM, definitely I'm an ECM by the definition, but when I talk to graduates who come into the company and they say, how did you get to where you are? And I go, look, I just so happen to maybe if there's no shame in potentially someone saying, here's an opportunity, would you be interested in it? So would you share the opinion that if an opportunity comes your way, there's absolutely nothing to be ashamed of going, I took opportunities that people gave to me.
Yep.
And you don't have to fight for it all yourself, because I see that a lot in young graduates who are like, no, I need to prove that I can stand on my own 2 feet, rather than accepting help that may be there, and certainly that element of luck of someone opening a door for you because they just like you as a person.
Yes, and a lot of these collaborations that I've developed have been people that have introduced me, and looking back at it now, actually, my PhD supervisor and somebody who's still a mentor to me, he introduced me to a lot of people. So I say it's luck, but I've kind of had a mentor who's passed those opportunities on to me. And now as a head of group, I try and do that for others. And I actually really enjoy doing that for others. So as an example, I was approached about doing some consultancy work, and I simply just didn't have the time. And that was estimating irregular migration in Africa. So it's building on some of that hidden human population work. And really, I wanted to be able to say yes, but I knew fundamentally there wasn't. So actually, a colleague of mine within the stats group, I asked him if he might be interested. He hadn't done any of that type of modelling before, but it brought some time out for him to be able to sort of learn about it and then interact with the institutions over in Africa. So I would honestly say that now as a mentor, I like giving people those opportunities. So absolutely, you should not feel bad about taking them whatsoever. And really, somebody who is supporting you to be able to develop your career, you're then going to be paying back to others. So it is very much this cycle that as you kind of go through your career, you do kind of take on that leadership role, and then you see, oh, that's what worked for me, and that's what you can then pass on to others. But I am incredibly fortunate to have had good mentors, and I know our university is developing it more, and it's certainly something within the statistics group that I've implemented. So we were lacking mentorship for a long time within our group. So I think it's really important to have those conversations with sort of academics at all different career stages, and to kind of get an idea of how have you made it? And, I say luck, but you do have to be selective with what you say, and you do have to look at it. And sometimes you have to be quite blunt with, okay, well, is that going to be useful for me? Am I going to find it interesting? Or actually, is it another thing that's just going to add to my to-do list, and it's not really going to give me a lot back? And at that stage, you need to think about maybe you should say no to that.
Yeah, that's great. Thank you very much. Like I remember watching or reading. I think it was reading an article, and it was someone who basically said anyone who says that they've achieved everything themselves is fundamentally lying.
Yeah.
And I think that's a huge lesson to learn, that, you know, because obviously sometimes I look on LinkedIn, people are like, I've done this all by myself, and I always think, no, you didn't. Maybe 1 in a million people might, but I hope if there's anyone on the phone on the phone. Wow, that's showing my age now. Anyone on the Zoom call who, you know, feels like they need to do everything by themselves, get yourself a sponsor, a career coach, a mentor, wherever you are, there will be a name for it. And you know, I'm currently sponsoring or supporting a young engineer with his chartered engineer application, and I've actually ended up putting some work his way that would fill in a gap in his capability, so he can learn and develop and work towards that. And I think there's a huge thing to learn is to go, if that comes along, if opportunities come along, don't be afraid to say yes, please, and recognize that and just pay it forward. So that was great to hear someone mention that. Fantastic. Thank you.
Thanks.
Good. I'm glad. So, I mean, I have more questions, but since this is being recorded, maybe I'll just ask you in the break. It'll be easier. And Ariana put a really nice comment in the chat, which is I'm going to take this opportunity to segue into her chat, into her talk. So Ariana is a PhD student at Brunel University in London, and she focuses on mathematical shape analysis and application techniques to real data. And if you look at the title of her talk, it's from monitoring endangered species to digitizing insect collections, which this is by complete fluke that I've scheduled it this way. I didn't do this on purpose, but I think they have this nice common theme, hopefully, that we're going to see throughout. She now works at the Natural History Museum as a postdoc, and I think we'll probably hear a little bit about that as well. But there was also something else I would like to say, that she just recently completed her Viva and passed. So please, congratulations on that regard. Welcome to the club. And please feel free to take it away now.
Thank you. I'm just having a little bit of video problem at the moment, as you can tell. Can you hear me though?
Yep.
Okay, so that's one thing. I don't know if anyone has any advice right now. It does say, it just says cannot start video. I'm guessing no one can see me.
That might be that whoever is the head has turned off video capability for others. So James, maybe you need to enable it.
I don't think I can do anything, unfortunately.
No, my video works too, so maybe it's not that. Although
Okay. Might just be my laptop then. I mean, I could exit Zoom and come back again. Oh, hold on, something's happening. Oh no.
Share your screen.
I can share my screen. Let's do that stuff instead.
Maybe that'll kick something into gear.
Yeah, maybe. Oh.
We can see your screen just fine.
Your screen.
Okay, that's great.
And I caught a sight of some Toy Story characters, so I'm looking forward to this.
Thank you so much for the introduction, and thank you for congratulating me on my Viva. Very glad that that's over now. Okay, I'm just gonna put this in full screen mode. Okay, can everyone still see?
Yeah, all good.
Okay, awesome. Thank you for having me. Apologies for not having a camera working. And yeah, so my name is Ariana, and I'm now a postdoc at the Natural History Museum. So I'm going to talk a little bit about my work during my PhD and what I love most, which is shape analysis and how we can use that to answer real world questions through various collaborations in the world. And yeah, and at the end, I might add a little bit about some work I do at NHM as well. So I get to share some exciting insects at some point. So yeah, the title is Shape Analysis, Imaging and Machine Learning, from Monitoring Endangered Species to Digitizing Insect Collections. Okay, so just a bit about myself first. So I was a PhD student from Brunel University, and my PhD was on the applications of elastic shape analysis. So this is the real world applications, and some people say, well, what's real world applications? Because applications are real world, but the difference is that in my area of shape analysis, a lot of the time people just work on some data set that's been given somewhere online, and they apply their methods to it, and that's the end of the story, and they call that applying shape analysis. But where my work differs is that I collaborate with people outside of my field in order to essentially spread shape analysis awareness, see how we can incorporate shape analysis and make it work for other people's projects in other areas. And right now I'm a postdoc at the Natural History Museum doing machine learning. So I do machine learning and imaging mainly, and yeah, it's very fun. And my interests are shape analysis, machine learning, imaging, and anything related to geometry and conservation I also love. So I absolutely love the talk that we had just now. That was awesome. Okay, so this presentation is going to be a bit weird. So I'm going to start by just giving a brief introduction. Then I'm going to focus a little bit about the math side of things, but I'll try not to focus too much, because then I want to go on to the application side, where I'm just going to introduce 3 or 4 projects, depending on how much time I have, that I did either during my PhD or as a postdoc here. So yeah. So as an introduction, what is a shape? Some people ask. Well, shapes are absolutely everywhere, and a shape can be anything you want, really. It's probably a good thing that you can't see me right now, because I'm getting very animated about shapes. I absolutely love shapes. So a shape can be, for example, an outline of an object, or it can be a growth curve, for example. That can count as a shape. So the Oxford Dictionary, that says it's the external form, contours, or outline of something. But where we look at shape in shape analysis is that we want to ignore the shape-preserving transformations, such as scaling, such as rotation, such as reparameterization. So for example, if you're picturing the shape of a violin, if you just scale that violin by scale factor 2, for example, the shape of the violin is the same. It's just the form that's changed. So shapes can be studied in various ways. In the museum, in a lot of areas outside of mathematics, in ecology and biology, landmarks are the most common method to use. I have a look mainly on curves, and in the area of elastic shape analysis, the focus is on curves and images. So I've just highlighted curves there because that's what my focus is on. So a curve, I just mean a mapping into usually R2 or R3. Okay, so shapes are all different, and they lie in this massive space. You're not in an easy Euclidean space here. You're in a much more complicated space. For example, if you've got contours around, if you're looking at points describing the outline of a mug, for example, if you reordered the points, that's still the same shape. So you're not actually in Rn if there are n points. You're really in R2n, and that's ignoring all other information about shapes. So what I'm trying to get at is shapes are very complicated. So shape analysis enables us to define the metrics properly to analyze the shape data. And that's the main part of it. So it allows us to define those metrics in order to study the shapes in their true space, not make any assumptions about data. A lot of the time when people study shape data, particularly my colleagues that I see in the museum, things like linear methods will be used even if your data isn't linear, but you'll hear all sorts of things about using PCA, for example, even though PCA assumes that your data is linear. So that's where shape analysis differs from all of that. And there are various applications, some of which I'll go through here. My favorites are the birds, because I love birds a lot. So that could be from analyzing growth curves to looking at the evolution of an object. For example, I work on a project related to the evolution of a Greek vase, and it relates a lot to medical imaging. That's why shape analysis pops up a lot as well. And yeah, there's a whole area of places where it can pop up. Okay, so a common framework, especially in my research, is first to get your data and process your data, because you might not have a shape at the start. For example, maybe you're only interested in the outline of a Greek vase, but you have images of Greek vases, so you need to get those outlines. So that's where image processing techniques become useful. And the middle stage, that's where we do a lot of the shape analysis. So that could be, for example, computing distances between two shapes to try and quantify differences, essentially. So how different is one vase from a different vase? How can we answer that question with just one value? And we can do that with tools from shape analysis. And that moves on to the analysis stage. So we can then put those values in to some sort of machine learning algorithm to answer other questions. If you're interested in classification, you can do classification on a distance matrix, for example, or it can relate to clustering and all sorts. So image processing is something that's definitely looked past when looking at applications of shape analysis. People usually ignore the image processing step and have someone do that for them. But this is such a vital part of any project that uses data. Data processing is one of the most important parts and most underestimated in terms of difficulty as well, usually. So when I talk about image processing, a lot of the time I'm focusing on image binarization, which is taking an image and making it into just black and white, having a clear image like you can see in that center figure. And the other things I work with is contour extraction, so finding an outline of an object, or using segmentation as well. So if you had, as you'll see in some other applications, if you had a picture of a table with various items on it, can you segment the things on the table? So can the computer algorithm find the kettle on the table or the toaster on the table? Okay, and that links to machine learning as well. So I pose a question here: how can machine learning be involved in shape analysis related projects and vice versa? And one way I already mentioned was image segmentation. So in the image processing stage, you can use machine learning to segment your image and then move on to shape analysis. So that's one thing you can do. Or it's what I was referring to earlier, which we can use machine learning after we've used shape analysis, essentially, which is if you've used shape analysis to compute distances, then can you put that into a machine learning algorithm afterwards? So as a post-processing step. An example of that is a KNN algorithm, which I might go into later if I get time. Okay, or we can embed a metric straight into an algorithm. That's another way you can combine shape analysis with machine learning. So for example, picture a k-means clustering algorithm, but instead of computing the Euclidean distance between the mean shape and your template, you can compute some sort of geodesic distance using tools from shape analysis. Okay, so quantifying shape variability. So I won't talk too much about it, but shape analysis essentially started from, or became very popular from Darcy Thompson's work from On Growth and Form. So that was all about being able to change one shape in order for it to look like something else. So he did this by drawing shapes onto a grid and then essentially stretching or bending that grid so that shape looks like another object. And in this case, it made one species of ray finned fish look like another species of ray finned fish. And that's kind of what our work is about as well. So we're trying to bend or stretch or bend an object so it can resemble another object. And that's why a lot of the time you might hear the term elastic shape analysis. It's because we're treating that as an elastic sheet. And we do this with diffeomorphisms. So a diffeomorphism is a map between manifolds. And I won't go into this too much, but I just wanted to show you this figure really, which shows how if you compose a diffeomorphism with a curve, you can change what the curve looks like. So the original curve on the left of the corpus callosum has changed into the figure on the right using diffeomorphisms. And these are super important things in shape analysis because they lead to the idea of geodesics. So geodesics are essentially the shortest path between two points. So if you're thinking about being on a sphere, on a globe, two people on a globe, the geodesic between those two people, so the shortest distance between those two people, will be the shortest arc on a great circle between the 2 points. So that's quite a nice example. So we're trying to find that shortest distance between points in order to tell you how different this shape is from another. So the way that works is we use these geodesics to morph the shape into another shape. And that can then that's then used to compute that distance value. So I won't go into too much detail about it. I'm just saying I'm giving out a lot of information right now, just because I'm really excited about the application side, which is kind of the main part of this talk. So we have one template curve and we have a target curve, and we want to morph one to the other. And essentially, we want to work out how much energy is related to do that morphing, and that's where the geodesics come along. Okay, so I won't go into too much detail about this at all, actually, but the important part of this page is the fact that the set of diffeomorphisms, so diff_R2 here, because we're working with 2D curves here, so that can be analogous to an infinite dimensional Lie group. And that then tells us two things. So first, it tells us that we're in some really complicated infinite dimensional Riemannian place, which means that computing anything on it is going to be rather complicated. So computing geodesics, computing other statistics, it's going to be difficult. It's going to be tricky here. And the other thing it tells us is that it has that tangent space that we can work on. So this brings me to this page here. So this diff_R2, this is a very complicated space, and if you want to compute distances here, it's going to be tricky. You can find algorithms that do this, and the main algorithm, if you're in the area of elastic shape analysis, is within the LDDMM framework, which computes geodesic distances on this complicated space. But alternatively, we can transform the space into something much easier, so we can compute distances much more easily. And that's kind of the beauty of shape analysis, that we can think of these things and do these things. So if we transform the space into a sphere, then computing geodesics, finding distances, is much easier, because all you have to do is compute the great circles. Or we can even go one step even further and just transform the space into something linear, and then you just have a simple Euclidean distance between 2 points. So I won't go into details about this, but the method on the left, which makes things linear, there's a method that can do this, which is from geometric measure theory, and it's called geometric currents, which I might go into at some point, but it depends on time. But the more important one, which is my favorite part, which is the SRVF, so square root velocity function representations, and that lets us transform the space into spheres, which I will go into slightly more detail about. So square root velocity function is essentially what it says on the tin. And the beauty of them is that when you're dealing with absolutely continuous curves, your space has been transformed into L2, which means you can use the L2 distance, which is very nice and easy, and not some complicated distance metric. And that's the beauty of SRVFs. And as this is not a geometry talk, I won't go into too much detail about. I've just added them here in case anyone wanted the slides later on, so they can look at it. Okay, so one thing I mentioned earlier was the fact that there's a difference between shape and form. So we are looking at the shape of an object, not at the form. If a violin is one size and you scale it or you rotate it, it still has the same shape. And there's also the question of, what about if curves look the same but are actually quite different? And by that I mean if they've been reparameterized differently. So these are two things that we need to sort out. We need whatever distance metric we use to be invariant to these things, or we just need to find some other way to ignore them. So what can we do? So we can remove these transformations in advance, which is a common thing people do, especially in biology. You may have seen people use precocities analyses to make everything the same orientation, for example, and scaled similarly. So that's one option. Or we can use some sort of representation for the curves that's invariant to shape-preserving transformations. Or we can quotient out the transformation groups from the actual space. So if we do that, if our space was L2, we can quotient out the reparameterization group, which here I've just used gamma here. So these would be other reparameterizations of the same curve. So if two curves have different reparameterizations, we want to essentially ignore that. And rotation as well, which is why we use SO2. So now we've got a quotient group, and this is important because it tells us that our shapes are actually equivalence classes, and that's quite fun actually. So this is very helpful because now we can work on the pre-shaped space. So if you look at the quotient space, we can work on the L2 part here in order to find a distance on the actual shape space. So on this SSRVF that I've called it here. And we can work on the tangent space to compute things. There are many methods that allow us to use this framework to compute distances between shapes or to morph shapes. A dynamic programming algorithm is the most common way to do this, most popular way to do this, purely because it's quite speedy and easy, which I won't go into too many details about. And another method is called SRVF parts training, which I won't have time to go into, but it's a very good method. So I've labeled it here in case anyone wanted to find out more about it. And here's just some examples of what we can do using these methods. So we can align curves together. So if you look at the top left plot here, we've got two growth curves from, I think those are from the Berkeley growth data set. So they'll be of children data. And in the bottom two curves, they've been aligned. In the bottom two plots, the curves have been aligned using various methods within this framework. So various methods of matching curves or morphing one curve into another. Okay, so I don't think I'm going to get time to go through any of this, but here's just another example of a plot where you can see one vase being transformed into the other. Okay, so this is the main thing I wanted to talk about, which is the application side of things. So there are loads of applications of shape analysis, and it's quite fun to do, and we're just trying to spread the word essentially because it's not used outside of shape analysis much. So one project we were working on was with a bunch of different people, so historians, archaeologists, malacologists, botanists. We all came together and thought, can we use shape analysis to classify these objects just from their outlines? And not only can we do that, but there are some other questions involved here as well. So are our methods better than current methods being used in these areas? Or are these methods, can they be a human, for example? Because in a lot of these areas, humans do classification themselves. A lot of archaeologists, especially if you go into ancient Greek vases, classification is done by eye. They will either hold the vase in their hand or look at an image of a vase. That's a very common thing that's done. And we wanted to see how shape analysis algorithms can compare and how other algorithms can also compare. So as I said at the start, image processing is super important. And this project involved a lot of that. So we wanted to take an image of an object, create and extract the outline of it. And the first step to do that is to threshold the image, because if you're just extracting an outline from the original image, it's likely that you're not going to get a good outline if your image is noisy. And when you're dealing with pictures of Greek vases that were taken 100 years ago, that's all the time. It's not nice data at all. But when you get complicated shapes in general, it's not easy to do any contour extraction without having thresholded. So thresholding is just another way of saying binarization, essentially, in this scenario. Okay, so the other steps that were involved here are smoothing. So we want to smooth the curves. So when you get an outline from an object, like you can see from this cup, this Greek vase, the Greek cup at the bottom, we want to smooth that curve as well. We don't want it to be noisy. And that's something we do with everything. And the other thing we want to do is additional alignment. So as I said before, our methods, we can make them invariant to shape preserving transformations. So we can quotient out SO2, for example, or the root parameterization group, or we can specify curves to be unit length, or specify use a derivative to get rid of translation variability, for example. But traditional methods that are used, especially in biology, their methods don't do that, which is why some pre-processing is needed, such as Procrustes alignment. So we do that as well in order, because we're making a comparison between other methods, it's a fair test if you do it that way. So if you just align all of them in the same way before going to the next step. So the next step is actually to compute distances between shapes. So we've got all this data, if I go back here, of vases, leaves, and shells. So there are about 700 vases and 400 leaves and 200 shells. And we treat them all separately. We don't mix the data. Everything is done separately. Our aim is to compare all the objects within each other. So we'll do pairwise comparisons of all the vases, pairwise comparisons of the leaves, and pairwise comparisons of the shells. And we do those pairwise comparisons using shape analysis methods and using non-shape analysis methods. And one of those non-shape analysis methods is eigenshape analysis, because it's a traditional common method used outside of mathematics, actually, especially in museums and biology. So once we've computed those pairwise distances, the next thing we could do for classification is to use machine learning. And a KNN algorithm goes perfectly with that, because KNN only requires a distance matrix. And that's what was outputted in our previous step, where we use those different methods to get the pairwise distances. So this is the best thing that we can use here. So we use a KNN classifier using various values for K, and we plotted the results to compare our methods. And we found that the shape analysis methods, the SRVF shape analysis method was the best one. So that's SRVF path training, which I didn't talk about. But that beat the other methods constantly. And this was through a bootstrapping test. So if you're thinking, oh, but it could depend on your training data, but it didn't, because we bootstrapped it 100 times, actually, which meant we used 100 different training and testing variations. So these results were really interesting, and they essentially encouraged us to continue using shape analysis methods. The purple is a different shape analysis method, so that's geometric currents. The orange is the LDDMM method, which was, if you remember, the method that works directly on that complicated space. And that creamy color, that's the traditional method that's used in biology and botany, etc. Okay, so other examples of how our methods were better was mean examples. So in the top row, we've computed the Euclidean mean here. So this was the cross-sectional mean of the leaves, just in this example. And in the bottom, we computed the Kakapo mean. I will go into the Kakapo mean in more detail soon, but that's essentially, you can think of it as a shape analysis version of the standard mean. And the means using the Kakapo mean are much, much nicer. In the top row, they barely look like leaves, some of them, and they look more like lemons and cabbages, whereas in the bottom row, they do look like leaves. So it's done a much better job there. And this is just another example as well. So yeah, we found out when we compared with the experts as well, which was when we just gave these experts images of, not images, the outlines of the vases and the shells and the leaves, and we told them to put them into groups and classify them. And some of them did a very, very good job, but we found that the algorithms always outperformed the experts, especially the shape analysis methods. They came out on top every time. Okay, so now I want to go on to a second application, which was related to Kakapo. So this was a really cool project that I got to work on with the Kakapo recovery group in New Zealand. And the aim was, can we use shape analysis to monitor Kakapo? So if you haven't heard of Kakapo, definitely Google them, because they are one of the best animals in the world. They're these adorable flightless birds, and there's only 200 or so left in the world, and they are endemic to New Zealand. So currently what they do is they look at the mean of the chicks, when the mean of the growth curves of chicks, in order to monitor whether the chicks are doing well, if they're healthy or not. And if they're not healthy, they will take care of those chicks. So them into a different nest, or someone from the Kakapo recovery team can get even more involved and hand rear this chick. And the reason we do this is because these are very, very vulnerable chicks, and they definitely need help, which is why we wanted to see if we can create a tool that can monitor the chicks. So they're weighed very regularly, especially when they're first born, but we look at them from 0 to around 80 days or so. So that's before they've fledged, which means before they wander off by themselves into the world. At the first 70 days or so, they're usually with their mum or adopted mum, essentially. So we split the chicks into various groups based on birth or the year they were born, for example, and various things. And just to note that we didn't do that splitting, that was from the Kakapo recovery group. These are all very collaborative projects. It's not the mathematicians deciding whether we should split by the birth year. It's the conservationists telling us to do these things. And that's the beauty of it all, where we can work together and find these solutions. So yeah, currently the growth curve, the weights of these chicks are being monitored, and they're being compared to the standard mean. And you can see a little plot of some chick weights there and the growth of them just growing up. And they're quite adorable little things over there. And okay, I'm just gonna, oh, yes, they are very smart, and they're also my favorite birds. And I say hi to them every time I walk past the stuffed one at the Natural History Museum. Okay, I said we'll go back to the Kaka mean. So the Kaka mean is like it's like a normal mean, you can think of it. So in the standard mean, which you're probably more used, in general people are more used to, this distance metric here, this can just be or xi minus mu or something like that. So what we do with the Kaka mean is we just replace that distance metric. So that could be a geodesic distance. So if you're morphing, so it's slightly trickier because I skipped all those slides, but just you can think of it as the distance metric in that shape space. So whatever you define there, that's what we're calling our distance metric, and that's what we're using in the Kaka mean. But that's not too important right now. So I just wanted to show you these plots really. So on the left, we've got an example of how monitoring is currently done on Kakapo chicks. So the dotted red line in that left plot, that's the mean minus 20%. So right now, that's how they do it. They look at either minus 20% or minus 15%. And if a chick is severely under that, then they will look into that chick. They will get that chick out of the nest and do an inspection and see what else they can do. So our idea is instead of doing that, we'll compute the Kaka mean instead of the standard mean, and we'll create boundaries up and below this. And anything outside of the boundary is deemed unhealthy. So one thing I should note that when you compute the Kaka mean, your y values may get slightly messed up. It doesn't hold it doesn't maintain those y values. So if a chick's curve, once it's been morphed into another curve, so once that curve has been aligned to this Kaka mean, its y values aren't as helpful when looking at it in a plot like this. And in the right plot, we've got aligned curves plus the Kaka mean. If the aligned curve is above a Kaka mean, that does not mean, sorry, above a boundary, that does not mean that the chick is necessarily overweight. That's why we need those boundaries there. It just means that it's very far away from the Kaka mean, which is why we look at the upper bound and the lower bound, not just the lower bound. So you might be asking, well, how can you tell if something is under or over a curve? Well, we tried a lot of various things with this, and the solution we came up with was to look at consecutive points. So how many consecutive points are underneath or outside of this boundary? And this is just an example with two curves. So that's Aria and F2 2002. And also just to note that you might think some names are slightly weird, but that's because they don't get their name until after they've fledged. So Aria has a proper name, but F2 2002 sadly passed away, so it did not ever get a normal name. Okay, so here we just have an example of how a curve can, so we've got two growth curves here, and we're going to count the number of consecutive points that it's outside of the boundary. And we use various optimization techniques to work out what that optimal number was. How many should we look at? How many points outside of the boundary should we be looking at? And in the end, we created this algorithm that can monitor these chicks for us and make a prediction based on that. So it looks at the weight of the curve, compares it to this Kaka mean. So it compares an aligned, so you get the weight of the curve, you align it, and you compare it to the Kaka mean, and you see if it's outside of that boundary that's been created by the Kaka mean. And that was compared to the original method, which is just to get the original growth curve and the normal mean, and just to see if it's under or over. So in those top two heat maps, sorry, you've got the original method, and in the bottom two, you've got our method. So the only important thing here is just to show that our method got much better scores. So we computed an F score here, an F beta score, but our main concern is with not misclassifying unhealthy chicks. If you're misclassifying a healthy chick as unhealthy, it's bad, but it's not as bad as misclassifying an actually unhealthy chick that can potentially die. So our F beta score was based on that. So we made it more towards that. We based it on that essentially. So we created our own score based on that. And when you do that, you find that our method works really well. So the top on the left. So if we focus on one heat map. So let's look at this this bottom right one. Sorry, I don't know if you can see my mouse. But we've got these chicks. So these chicks are the chicks that were actually unhealthy, and our algorithm predicted them as unhealthy. And these 5 over here, these were actually healthy, and our algorithm correctly identified them as healthy. So it did a perfect job at predicting the health of these chicks. Whereas if you look at the top one, the 3 of the unhealthy chicks were classified as healthy, and 5 of the healthy chicks were also, but 5 of the healthy chicks were classified as healthy, so that bit was fine. Okay, and in the end of the day, we made a Kakapo tracker for them. So the Kakapo recovery team now have this tool with them where they just type the name of the bird, and a growth curve, an aligned growth curve is shown with the mean, and a prediction result is shown as well. And that's what we're working with the Kakapo recovery team. Okay, so another project, if I have time, hopefully, was one I really wanted to talk about, which is to do with kiwi birds. So this was a really exciting application of shape analysis because shape analysis has not been applied to sound data before. So you might not think of sounds as images, but if you plot a sound on a spectrogram, you're going to get curves. And you can see a spectrogram here. That's an example of a spectrogram, and that's an example of a kiwi. So can we use shape analysis to monitor kiwis now? So the idea is not to classify a kiwi from a different species of kiwi, but the idea is, can we do classification within a species? So can we classify an individual kiwi from another individual kiwi? And that can then be used for in situ monitoring and other tools such as that. And this is something that other institutions are looking at as well, including NHM and ZSL, so London Zoo. This was in collaboration with avians at Victoria University of Wellington in New Zealand, hence the iconic New Zealand bird, the kiwi. So what we did here, so our first step was to process the data, again, going back to image processing, to get those curves from the spectrograms. And the next step was to pairwise register these curves and then use machine learning to do the classification. Do I have time? I don't really remember what time I started, but I feel like I started later.
Yeah, you have plenty of time. Don't worry about it.
Do I have, what, 10 minutes, do you think?
Yeah, of course.
Okay, perfect. Awesome. Thank you. Okay, so sound data is very, very messy. And we want to get those curves from the sound data. So if you just use image processing techniques here, it's going to be quite difficult to get those curves just because your data is naturally very noisy. And there are lots of people working on denoising these spectrograms. A lot of machine learning, especially people doing wavelet-based things and more signal processing related things to do this. So what my area is in, what my research was, is whether we can combine signal processing with image processing in order to extract these curves. So I don't, oh, sorry. Okay, sorry. Okay, so one way we did this was we computed the fundamental frequency. So that's something quite well known in signal processing and with audio data in general. The fundamental frequency is usually the lowest dominant curve. So that will be the noise that you're mainly hearing when you hear a sound. And harmonics are then produced from that. So harmonics are just variations of that fundamental frequency. So this lower curve over here, that's the dominant one. And everything on top of that is just a variation of that. So by finding this fundamental frequency using tools from signal processing, such as if you're in the area, the yin algorithm, for example, we have a rough idea of, we can find the fundamental frequency. From there, we can then find the next fundamental frequency and then the next one. So we can get, in this case, we looked at 3 levels. So we used that in the next stage, which was when we were doing image processing, we then told the algorithm to only look at those specific places where those fundamental frequencies were found. And this really helped us because it enabled us to extract these curves. So I don't know if you can see them very clearly, but I've highlighted some in blue and some in red here. So these are curves taken from a spectrogram of a kiwi. And if you're interested in which kiwi, it's the North Island brown kiwi in New Zealand. So the aim here was to compute the distances between these ridges. So between these harmonics. So because there are multiple harmonics, what we wanted to do was do some sort of distance between those 3, in this case, those 3 curves all at once. And in a more mathematical sense, that means we were interested in finding the distance in a product space, which is quite tricky to do. So instead, what we went for was a more pairwise approach, which was to grab 3 from, so we grabbed 3 contours, so 3 curves. So in each segment, we looked at 3 curves. It's, yeah, I don't know if you guys can, oh, if I can zoom in, you can see they're everywhere. Okay, so we grab all of these. Oh, sorry. Okay, so we grabbed these and we compared it pairwise. So the lowest curve will be compared with the lowest one, and the middle curve will be compared with the middle one, top one with the top one, etc. And we did this for all the trios, which I call them here, all the trios we found from the sound data. And once we did that, we can build a distance matrix once again. But there are other things that we might want to look at when comparing sound data, such as the actual, the median frequency, for example, the length, and other information that the conservationists, for example, can be interested in. So we found a way of combining that information with the shape of these curves, with the information from the shapes of the curves. So it wasn't just the curves we're looking at, we did look at additional information such as that median frequency. And eventually we went back to using a KNN algorithm, which was then used in a further optimization algorithm to create essentially a meta distance matrix that can be used for classification. So that was just one project that we worked on. So now I'm gonna, if I've got time, briefly talk about some work I do at NHM. So at the Natural History Museum, we've got a lot of data, as I'm sure you're all aware of. And in that data, there's estimated to be 25 million pinned insects. So if you see a tiny dot there in this camera, that is a pinned insect. So the aim that the Natural History Museum has is to digitize all of these specimens. So take a picture of all of the pinned insects, so it's readily available online for researchers to use, but also to capture the label information from them. So these insects are on a pin and they have a label on them. So at the moment, a digitizer will take a photo and then they'll write down what it says on the label themselves. And this is going to take a while for them to do all 25 million specimens. So the question is, can we use machine learning, image processing, shape analysis to do this for us automatically? So this device was made called Alice, angled label image capture equipment, where we've got multiple cameras pointing at a pinned insect, and they take photos all at once from different angles. And we want to use this to automatically extract label information. The reason why there's multiple cameras is if there was only one camera, the pin might be in the way, or the insect might be in the way, something might be in the way. So it's better to have multiple cameras here. So what we want to do is find the label from these images, and then we want to combine those images, which will have things blocking them, in order to grab a clean image. And then at the end, we want to transcribe them, which means we want to extract the information on that label. What does it say? Who's the collector? When was it collected? Where was it collected from? And that then goes onto our database. So here's a better example of a pinned insect. So here we have some sort of fly on a on a pin, and we've got the label information here, so these rectangles. So these are the information that we want to get. So originally we thought, can we just use image processing to do this with no machine learning? And one way we tried to do that was to find the pin, because if you find the pin, you can work out where the pin is being separated by pieces of card, and that can tell you where the labels are. So that was something that we looked at. So this was part of a pin recognition algorithm that we created based purely on image processing techniques. And if you're wondering what kind of techniques we use, well, things such as the mode color, very basic thing, but the pin is usually gray. So we can look at the mode gray color, where in this image do we see most of that color? And that's roughly the technique that we used in this example, for example, to find the pin. Okay, but this isn't gonna do. You've got 25 million specimens, and you need something slightly more robust, and we turned to deep learning for that. So in this, what we actually turned to was a CNN approach, which I won't go into in this talk, but essentially it allows us to grab the label information to do segmentation. So it finds the labels. Note that this doesn't transcribe the labels or anything. It doesn't even rotate the labels. All it does is detect whether a label is an image and where is it in that image. And it pretty much does it by magic. It's very, very neat. But we've got some other tasks left to do. So once you've got that label, we need that label rotated, for example. This is where you go back to geometry and you do things such as perspective transformations, which I, again, won't go into detail about. But it's just to show you the kind of work that it involves. So once you've rotated it, then you can go to the stage of merging them with other labels and eventually transcribing them. So where shape analysis might come into this, for example, is once we've rotated the label, so once it's been, once we've done that perspective transformation and got it to this bottom plot here, some of the time the label might be upside down, and that's quite hard to tell when this has happened. It's hard to tell automatically if something is completely upside down. So one way we do this is we actually extract the letters from the label. So we extract the contours that represent the letters or the numbers, and we compare this to a template that we have of whatever language it's in. So if it's in English, we've got the Latin alphabet, and we use shape analysis to, well, this is one of the things we're testing, is can we use shape analysis to compare these shapes. So compare the shapes of the letters on the label to the shapes of the letters in our template. And we find the closest letter, and from using and just focusing on that closest letter. So if it's found, if the smallest distance between all of this, all the pairwise distances is between, I don't know, the letter M here and M in our alphabet, then we just compare those two M's. And from there, we can use something like Procruste's alignment just to find the rotation, or using your other favorite method of alignment to see whether that M has actually been rotated. And that's just an example of how you're merging shape analysis, machine learning, and image processing to answer questions in the real world, do collaborative projects with people, and to try and find solutions and things. And that's the main aim of my talk and the main aim of my research. So thank you. Sorry for going slightly over and not having a camera.
Thanks a lot. That's a lot of content that you've gone through.
Apologize for that.
the applications. So this is like what's so amazing about this type of work with machine learning in particular. You've got this very typically hard mathematical notion in terms of like Riemannian metrics and shapes and this classification, then using it in a manner which is like that you work at the National History Museum. This is what's so fantastic about maths, is this ability to be able to take these things and work in many places. So
what I love about it as well.
yeah, it's just and it's really frustrating when, you know, when if you ever get to do outreach and you're like, oh, maths is just everywhere, no one believes you. So this is a great outreach talk. So does anyone have any questions? I know like we have gone a little bit over, but I as usual I have questions, but if anyone else has any questions first.
Yeah.
Oh, Rosie.
I have questions. I don't know if they're stupid questions, but I'm going to go ahead and ask them anyway.
No stupid question.
So one of the questions was about the last project that you were talking about, the Natural History Museum. So have they managed to automate like putting, getting the bugs and like putting them in, or like is that all still going to be done?
Such a good question. That is a very good question, because only 2 days ago we were talking about that. And at the moment it's a human doing the putting it in there and taking it out. But we're actually working on writing a proposal to get robots to do this for us. So then once that's happened, then everything will be completely automated, apart from turning a robot on and off maybe.
Yeah. But I don't know if it's exciting and a bit scary.
Definitely cool, thinking that I might have robots in my office at some point.
Yeah, but what if they go wrong? Evil robots. And my other question was about the classification of bird calls, and I wondered if you would be able to use the same kind of methods to like classify the type of a call, like whether it would be like an alarm call or something else. Like, is that a potential like application for it as well?
Yeah, that's definitely a thing. I think it might depend on the type of species. So I know with some, going back to birds, I know some birds like kiwis or moorporks, the type of call that they're doing, whether it's to warn something, whether it's related to mating or whatnot, will look different sometimes in the spectrogram. And in those cases where their calls are different, then this can certainly be applied to that. But yeah, that really depends on the actual species. I can't say that will work for everyone, because sometimes the harmonics just aren't visible to us in the spectrograms, and sometimes it's not even the harmonics that do the differentiation. Yeah, so yeah, not all the time, but sometimes certainly that can be applied. That's a great question.
Yeah, so it would work for some species. I was even thinking as far as like potentially being able to assess like the health of a bird on like the type of calls it's using, and if it's a very angry bird, for example.
Oh, that I don't know about. Maybe. I mean, I might be slightly wishful thinking if it does that. That's slightly more difficult, but that'd be really cool, definitely. And it can, if that did work, that could certainly be used for the Kakapo as well, not just the kiwis and things. But yeah, and that sounds slightly more difficult, but potentially, yeah.
Thanks for the questions, Rosie. Does anyone else have any more at all? Okay, I think that's no. So let's take 15 minutes then. I'll ask my questions at some other time, as it's being recorded. So hopefully Ariana and Rachel will stick around for a bit so I can ask some questions. But feel free, everyone else, to sort of take 15 minutes, have a refreshment break, and see you back soon.
Awesome. Well, I'm going to stick around for a little bit, but then I have to head off because I am at a premiere in that will eventually kick me out. So,
okay.
But what I might do is I might leave the Zoom meeting and come back, because I think that might be why my camera isn't working.
Yeah, that's fine. I'm happy.
So if I leave, would I be allowed back in?
Yeah, I'd assume so.
Okay, awesome. So I'll see you in like 30 seconds.
Yeah.
It's been some interesting first talks.
Yeah, very, very good.
On a completely different subject, has anyone seen the last question on the Edexcel GCSE higher non-calculator paper this year? A friend of mine who's a teacher sent it through to me, and I timed how long it took me to do it, and it took me 15 minutes. So, and it's meant to be 4 marks. Now, I just feel that's a bit unfair. Anyone got any questions they might want to ask of the ECM committee that's not necessarily relating to a talk?
Ah, now we see you.
I have a face now. Yay. I don't really know why it didn't work previously, but at least it's there now. But thank you so much for having me, and I'm sorry for doing an incredibly long talk with lots of content. It's because usually I do one or the other, whether it's applications and a little bit of maths, or maths and a little bit of applications, and I was trying very hard to shove everything into one talk.
I'm a big fan of the speed run, so like, I do the exact same thing. So
it was very good.
I want everyone to know everything, and so I'm just like, blah, blah, blah.
Exactly, and you can always send the slides to people so they can get the actual content at some point.
Let me ask the question that I had, which was, so what sort of tools do you use? Like computational tools? What's the sort of like time efficacy for these things to sort of run? So when you're doing your shape analysis, do you have like a particular set of programs you use?
So I use Python for everything.
Okay.
For the main shape analysis stuff, I think it depends what we're doing. A lot of the time, I talk about SRVF path straightening very quickly, but that's something that we use when it comes to closed curves. And the library we use for that, I'm going to write it down in case anyone's interested, is called FDA SRSF. So that's the library, Python library. That's quite useful for people. And there are MATLAB things as well. There's libsrvf for MATLAB. I don't know if anyone is interested in this, but just in case. I'm
stealing this for a maths tea. So for the maths tea on coding soon, so I was like, yeah.
No shame, have you, James? No shame.
Oh gosh, but yeah, I use Python for everything. I avoid MATLAB when I can. Sorry?
And the visualization as well. Okay.
Yep. Yeah, even if it's just to make an image slightly smaller, crop an image, I use Python for absolutely everything.
Yeah, I remember watching a video about there are programs that do a lot of there was one in particular I was watching, which was like they had like a picture of a rabbit from different angles, and you could basically use the interface to be able to select where you think it's roughly going to go.
Yeah.
I just thought if you have anything like that, then
no, I tend to there are libraries that can do things like that, just as you said, especially in related to the what side, the contour extraction side, where you have to manually fiddle with things. And because we want everything to be automated, we avoid any of those programs because our aim is automation. And essentially what we want in life is, if someone said, I have this data set, whatever it is, if it's bird noise or if it's images of kettles or something, we just want them to put their data set, put the file path, the folder where their files are, into our Python program, and everything else can be done for them. Contour extraction, machine learning, shape analysis, and everything like that.
Yeah, it makes sense. I mean, the fact that you've got 25 million insects, if you have any sort of human element in that, the whole point is somehow redundant. So it makes sense. Yeah.
Exactly. And that's why some people avoid this. They're not outside of mathematics, and they've got a lot of they definitely avoid this. A lot of the time people work with landmarks because it's easy to just point landmarks everywhere, rather than having to measure it properly and drawing the curve. So the beauty of these things and automation.
Yeah.
I think there's a question there from Rachel. Thank you so much, and no worries. How did you get the role with the naturalist? Did you contact them? Yes. So I actually applied for a different job at the Natural History Museum, which was to do with, I think I was more focused on just the imaging side of things, purely because I wanted to talk to them. That was my way in. So I had a really nice interview with them, and we spent ages talking about it, and I was incredibly excited afterwards. And in the end, I didn't get that job, which wasn't surprising, but they all still really liked me. So we just continued talking until I finished the PhD. And then once I finished the PhD, there was a postdoc ready for me that was more relevant to my area. So a lot of luck was involved with that, but I had really nice people in my interview that I got along well with.
Yeah, I was going to ask that as well. That's quite useful.
Yeah. Do you ever collaborate with the Natural History Museum, Rachel?
I think you're muted, Rachel. or at least we can't hear you. The perils of Zoom.
So it seems you're unmuted, but we cannot hear you at all.
Yeah.
Can you hear me now?
Yeah.
Oh, yeah.
Yes, there we go.
Okay, yeah, sorry. It must have been me shouting at my children that stopped my Zoom from working. No, I've never collaborated with them before, but I'm really pleased to hear them kind of engaging with kind of machine learning and kind of really modern methods, which, you know, I think so many organizations are now. And it's really interesting to see where kind of mathematicians and statisticians kind of end up on postdocs.
Yeah.
It really is so much more varied than it used to be.
Yeah, definitely. I think people at Brunel were always also slightly confused as to how I'm doing a postdoc at the Natural History Museum. I think they didn't know it was related to geometry or something. But yeah, that's definitely the beauty of it. And yeah, NHM, Q is doing similar things as well. And God, some other institutions as well are doing similar things in other countries as well.
Yep.
But yeah, it's really cool. And I loved your work as well. It was super cool. I am very, very biased, but it was really cool. I just love anything to do with using mathematics and statistics with anything to do with ecology and conservation. That's something I absolutely love. How did you get into that? May I ask?
It was purely by fluke. So actually, when I was in school, I thought I'd want to be a vet.
Oh.
And then I did some work experience at the vet and realised I'd absolutely hate this. So then I was like, well, I like maths, so I'll do a maths degree. And as I say, I didn't really have any idea what I wanted to do. And it was just sort of seeing some examples of how statistical models have been used in ecology during my master's. I thought that sounds interesting. And then Byron at the time, he had a project. So it was back in the days when you kind of got specific research council funding to do PhD studentships. And he had this project that was linked with France. And I thought, oh, great, I get to spend a bit of time in the south of France and go and study flamingos. And actually, I never did a single analysis on a flamingo data set.
Well, I can send you flamingo data from NHM if you ever want to.
Just putting it out there, if anyone still needs someone to volunteer and go do some data analysis on flamingos in the south of France, I mean, I'm not an expert in that field, but I will take one for the team.
Oh, God. That's amazing there. I love it. I think mine was also quite random because I signed up to do a PhD in geometry in shape analysis, but more on the geometry side of things, so the pure math side. And then I had this weird thing where my supervisor left, and the supervisor who then took charge was in New Zealand. And that changed everything because he's also obsessed with New Zealand birds. And so suddenly I got flooded with New Zealand bird data, which was the best thing ever. But he also kind of bribed me and said, if you work with me, you can come to New Zealand and work with Kakapo. I have not worked with Kakapo yet. I was supposed to go when the pandemic hit. Sadly, that did not happen. So hopefully next time, if we do meet each other, you would have worked with flamingos and I could have seen the Kakapo in person as well.
Yeah, I do have this wrong though. So my PhD student and my postdoc, my PhD student's currently out in Mauritius, my postdoc's shortly going to Mauritius, and I've never been. So I definitely haven't got my priorities right, I think.
So you're the supervisor, you're the one who gets to delegate the work, and you're giving, I mean, I know you said you like to encourage others to develop and all that sort of thing, but there's definitely something that you've missed there. I'm not saying deny them the opportunity, but maybe they need some supervision whilst they're out there.
Yeah, I think my husband would agree as well, actually. Especially if there's, if you need any support, I can come out with you.
In fact, if there's, this is a completely unrelated thing, nothing to do with maths, but my best friend, his partner's a archaeologist and doing a lot of work out in Senegal at the moment. And last year, basically, she managed to get funding for him to go out there for a month with them to be their driver. All he had to do was drive the 4x4, and he got to spend a month in Senegal. Bearing in mind, this was in prime COVID lockdown, and he was self-employed and basically just was earning nothing and he got paid to go to Senegal for a month. So
I'm
it's
in the
definitely
wrong
possible.
field. I'm in the wrong field.
It's
That's so
Dude, what are you
amazing.
talking about? We got to go to Luga Baruga. We
Luga
got to go to
Baruga.
Lough-
That's
we
the
got
one.
to go to Loughborough.
Oh, okay. Oh gosh, amazing. But I can't believe you haven't gone, Rachel. My supervisor always goes and, you know, just gets kiwis out of burrows. He delegates those tasks to himself. He's the one traveling.
No, absolutely. I should do. I think while my children have been young, it's been kind of much more difficult to travel. So before we had kids, like all of the conferences, I went to Brazil, I went to New Zealand, and so much fun. And over the last kind of, well, 5 to 9 years, really, they're kind of just more difficult to do.
Yeah.
And all of these exciting trips really happen kind of when it's still school term time. So just when the pandemic hit, my husband and I were due to go to Sydney on a work trip for me. And so we had it all planned. We had my parents coming to look after the kids for 3 weeks. And then COVID happened and we couldn't go because it was cancelled. So
That is
at
gutting.
some point we will resurrect that offer of 3 weeks of childcare and go somewhere cool.
I mean, I don't know if this speaks more of the quality of parent of one of my colleagues, but he seems to take every opportunity he can to travel to a different part of the UK and get a few days away from his 2-year-old and 5-year-old.
Very sensible.
To quote, he was like, honestly, sleeping in a travel lodge on a slightly lumpy bed is better because he gets to sleep throughout the night.
Yep. Although it's terrible now because I find myself waking up at 5 o'clock even when I'm in a hotel room out somewhere. So I can't even blame my children anymore. I think that's middle age for me.
I've got a sort of question relating to the image processing. So I've not directly done any using live cameras to identify things, but we often do, I mentioned earlier on, we do control points at work, but we're also now starting to be able to, the idea being that we could take a component and try and predict its degradation, there we go, by taking images of it and then running it through some machine learning. Or I say machine learning, running it through a computer to be able to give us an idea about how, not only what its condition is today, but how many hours worth of life has it got left before it might break.
Yeah. How well does it do?
Well, this was what I was leading on. Whilst I can't say exactly how well it does or does not do, have you found incredible amounts of frustration in how wrong those predictions can be and how difficult it can be to identify or even misidentify something?
I think yes. So when it comes to deep learning, definitely. So CNNs and things like that, which are the main things people use when it comes to machine learning and imaging. Definitely, there are lots of frustrations, especially when you don't have enough data. And that's kind of why I didn't want to do any deep learning on my other projects. I avoided that at all costs. There is no deep learning in my PhD at all, actually, because with deep learning, I have that. And it's usually because of data. You just don't have enough data. How much data do you have?
Last time we tried doing this, I believe we did it on 15,000 turbine blades.
Okay. Oh, that's quite a lot then. Okay.
Yes, but the interesting thing is that I think every single one of them failed in a different way.
Okay.
So, or so, although whilst it can look very, they can look the same on the outside, there's so many different ways in which they can fail. By the sound, because as I say, I've never really done a lot of it, but one of my colleagues has done an awful lot of this, and all I ever hear from him is, her, is just mass amounts of annoyance and irritation and why can't I just get this to work?
Yeah, I have that too. I think my main advice is to just not rely on it. So at NHM we are using deep learning for the Alice project, and it's only one step of it because we just don't trust it's going to be great. So we've got all these different algorithms that are used afterwards as post-processing to fix the errors essentially, and just because we don't trust that, I just don't like relying on one algorithm for one thing. It's always better to use multiple things to avoid errors like that happening, especially if you're using deep learning, you're gonna have some errors somewhere if you don't have enough data.
Yeah, I'd say this isn't my area of expertise at all, but the way that she described it to me was basically currently we do it by literally a human looking at it and doing a visual inspection.
Oh, wow. Okay. So how does the human look at it? As in, what do they look for?
They look for notches and chips out of, the metal, for example, look at if there's any cracks in there, try to identify key features, which we can then look at a chart and go, well, how long is that crack? And then from that, we can predict, because we know how long the crack is, we roughly understand how long it will then take to run all the way through or run to a point where it'll break. That's fairly well understood, but this process takes time.
Yeah.
Especially when, let's say in our civil business where, I don't know if I've mentioned, but I work for Rolls-Royce PLC. In our civil business, we have huge amounts of turnover on components as we overhaul jet engines. And obviously we want to continue using them as much as we can and minimize wastage. Whilst it both helps economically for the company, it also reduces wastage for the planet as well.
Yeah.
And being able to do that effectively, having them sat on basically a conveyor, the idea being is that it would sit on a conveyor belt and then just be passed through a machine which would scan them via cameras and images rather than traditional ways such as X-rays and CT scans, which is what we do now, which are very expensive. And that would then be able to pull out all the ones which then maybe go, these have these cracks in them that are this long, and it would automatically be able to automate this whole process. That's the basic idea that I can get from what my friend was telling me. But they are struggling so much with getting, basically she said, an AI to be able to recognize the difference between something which might be a thermal related crack or something which might be a stress CF load related crack. Yeah,
that's really interesting. That definitely sounds like one day it'll be possible. I think for me it just sounds like the AI side should be just use it as less as you can. Definitely use it because it's super great. It is useful, but just use the basic things as well. So for example, we use the deep learning sometimes just to get a rough idea of where the pin is. And then from there, you can use something as basic as the dark grayness is going to be the pin. And that's not machine learning. That's just that's what the human does. It looks for the gray pin. And it's something like that. If you can use your AI to do a rough location of where your crack is, and then use some image processing that's not necessarily AI to do those next steps, might be something that could be more helpful potentially. Essentially, use it in conjunction with other things.
Yeah, as I say, I can't comment on exactly how this is being done. I was just looking to try and get an idea of, because you said you'd had some issues with model fitting, for example, and false positives, false negatives. But I was more curious to go, what are the frustrations like in trying to get these to work to start with and recognize images and such? So it sounds as if it can be as frustrating or not frustrating as we want it to be, depending on how complex a technique we use, which basically just sounds like everything in maths.
Exactly. But if they ever want to have a proper chat with it, that's something. Yeah, I'll be really interested to hear more about it. That sounds really interesting. It sounds really cool. What's your colleague's name?
Unfortunately, I can't tell you.
Oh, okay. Well, if there's a paper on it at some point, then if you could share
as I say, unfortunately, working for Rolls-Royce PLC, we don't like sharing what things we manage to do well, and we definitely don't like sharing the things that we don't manage to do well.
Oh.
Well, it's the classic private sector problem. So there may be some stuff that gets published. I know that my colleague, she was doing a project in conjunction with the University of, never remember if it's Oxford or Cambridge, one of the two, on this sort of thing. So there's lots of things which are going off, and so I can see I will probably mention to her, say, look, I saw this talk at the weekend. It might be something that might interest you. I don't know.
Yeah, it could be cool. Yeah. Because I think it can definitely be done, and it will help you guys so much if it's right now a human looking at that. That sounds quite long.
Yes. Is there any more questions in the chat? Anyone? Oh, what's this about conferences? Conferences should be more accessible. In fact, that's a good point. I'd agree with that. Anyone got any thoughts on how conferences could be more accessible?
Hybrid conferences are always good.
be interesting
Yeah.
to see now that things are going back to in person, how people will go, what will happen. I know the IMA ran an in-person conference in somewhere in Scotland recently, Edinburgh, and apparently that was very successful in being in person. But I wonder if it's just going to be, we're just going to go back to exactly what we were doing now, if we'll have learned anything.
But the BAMC in Loughborough a few weeks ago was, well, they advertised it as in person with an online component or something like that. It was essentially hybrid, and it worked very well. I mean, I appreciate it took a lot of work from the organizers, but it worked very, very well. And for example, I was in several talks where the presenter was supposed to be there, but some last minute changes wouldn't allow that, and then they were able to give the presentation rather than canceling, as would have happened in the past. And I mean, the only thing that I would say didn't work that well was the poster session, but having a hybrid poster session is extremely challenging.
I hope that will not happen. Oh God. As in, sorry, the Metaverse conference in reference to the chat, not open stuff. I think the other thing to do with accessibility is costs and visas and things. Some conferences are ridiculously expensive, like talking £400 for some top ones. And some of them, that's not even including the people coming from all over the world who have to pay visas and pay for special visas. If conferences can do more towards that, that would help make it more accessible.
Well, and also sometimes there's a 5-day conference that you are actually interested in, maybe 1 day of talks, and you rather do that from your office than travel to the other side of the world.
We were discussing this the other day in the conferences meeting about there's two things that sort of came up. One of them was, whilst conferences can't be free, how ethical or how do you feel okay about charging someone so much money to be in a conference in their own house? And like there's like this toss-up of, you've got to pay for all of the like room, you've got to do room booking, the ability to be able to stream all of it, but does that mean that you should charge everyone the same flat fee and then add more on to the people in person for food? Is that fair and a justifiable fair use of resources? And the other one was I've forgotten. Edward, what did you say? Because that prompted my thought. What were you saying?
What did I say?
what were you saying?
The attending only part of it.
Oh yeah, and this is thank you. This is a good a problem that we have, particularly in the IMA, about industrial conferences, that people apparently we used to run industry events quite a lot at the beginning of this decade, and that this slowly people just started coming to it less and less because they had.
When you say beginning of the decade, do you mean like 2010?
Yeah.
Yeah, the beginning of last decade.
The beginning of the last one, yeah, sorry.
I know we all feel like it's still 2019 and we can't remember the last 2 years.
Yeah.
Sorry, I just wanted to clarify that.
And that, yeah, over the course of 2010 to 2020, it's I just declined because people couldn't justify the need for going into London to go to an event that they only needed 1 hour of talks for.
I would still do it for the networking side of it. I mean, if one thing I couldn't find any conference that make it work online is the networking, it's the coffee break, essentially. Or this, yeah. I mean, this is working quite well today, but this type of conversation in a big online conference is very difficult.
There are, like, we had the BMC in whatever year that was, used Sakoko, the platform, and that seemed to work all right because it gave you this ability to be able to go into virtual coffee rooms. Yeah. And so it bridged that gap a little bit. You can have the same thing with spatial.chat as well. You have this like spatial awareness of where people are, but it's just, whilst it is a step in the right direction, I don't think, I think it's folly to even attempt to try and emulate it. What's the point? You're never going to be able to do it. So like we should be able to, there should be a different, maybe the culture needs to change or something, but why try and emulate something which is quite definitely not.
Yeah, I think in the same way that you know, the pandemic happened at the right time for tools like Zoom that were ready but nobody was using, or well, almost anybody was using. I think the tools like Kakapo and all these other tools were not ready yet.
No. And they're so expensive.
Oh yeah.
That it makes it very difficult to justify. I think with that in mind, let's begin, because I realize we've gone over half an hour already, which is basically my fault. Sorry, Rosie and Jane, if you had to be somewhere.
You know, I'm missing the Formula One qualifying for this. So I am quite upset with you.
Just is BBC iPlayer or something?
It might be on my other laptop. But
I'm not gonna lie. I have a screen behind my laptop which may or may not have some cars going around a track on it.
I can see my husband currently watching it on his laptop, which is
I'm
very.
the only one not watching it.
James, you're missing out, mate.
Yeah. Well, I'm watching these talks because I'm a good participant.
Oh no, I'm watching the talks as well. I'm
Yeah.
doing what my fiance says I can't do, and it's multitasking.
I can't multitask. That's the problem. So.
But I'm not sure how good it would be to give a talk and watch the Formula One at the same time.
True.
Well, you can tell me at the end.
I have to dash now, but I'm really sad to be missing the last two talks. Where can I find the recording? Is that...
Good question. Not sure.
I believe they will be in the YouTube channel.
Oh,
Yeah,
okay.
the talks will make their way onto YouTube at some point. Whether or not it'll be the whole conference or whether the talks will be sliced and diced and put on individually, just keep an eye on an IMA page on the YouTube. No, an eye on the YouTube IMA page, and they should appear on there eventually. Turnaround times normally a couple of weeks.
Okay, perfect. Well, at least I'll get a chance. Yeah, I'm really sorry about missing it, but I just have to leave the hotel now. It's really annoying.
The Formula One.
I wish. I don't even know if it's done now.
It's not.
Oh. No, we'll have to watch that on our mobile phones probably. But thank you so much for having me, and I'm sorry for missing your talks. So sorry.
Thanks for coming.
Yes, lots of fun. Bye.
Bye. And I see Rachel also has to shoot, so thanks Rachel as well for staying for a while and having a chat. So let me introduce the final session. So up next we have Rosie. She's a research fellow at the London School of Hygiene and Tropical Medicine and is part of the Centre for Mathematical Modelling of Infectious Diseases, which you'll probably have heard of throughout the last few years. She's been working tirelessly providing results for the government, in particular contributing to SPY-M and SAGE throughout the pandemic on COVID-19 modelling. She's been utilising the model, which often leads to contributions to the ensemble, which are used by the scientific committees to make policy decisions. She did her PhD at the University of Sussex with me around the same time, and then moved into a postdoc in life sciences and is now working at the London School of Hygiene and Tropical Medicine on a project funded by the EU Horizon 2020 EpiPose project. So please, Rosie, feel free to take the floor now.
Thank you so much. Thank you for the lovely introduction. Let me get set up here. So hopefully this is showing full screen for you.
Yeah.
So yeah, as I said, thank you for inviting me. My name is Rosie Barnard, and yeah, as James said, I'm working as a research fellow in infectious disease modelling at the London School of Hygiene and Tropical Medicine. And my talk today is really going to be about some of the mathematical modelling that I've been doing in the last few years, and alongside that, what my early career reflections are, having done that work. So I'm going to start with a little bit kind of going back to a bit of my background and like what led me to where I am now. I'll briefly talk about some of the modelling I did before COVID, but mostly I'll talk about some of the modelling I did during COVID and what my early career reflections are. So back to the start. Before university, I wasn't really sure, like I didn't have a clear plan in place of what I wanted to do or be. But I really did enjoy mathematics, so it was a case of, okay, well, I'm just gonna carry on studying the thing I enjoy. And I think that's a bit of a theme really throughout my career so far. So yeah, I ended up doing an undergrad in maths, and then I ended up doing a PhD in maths, and I've done a couple of postdocs which have been slightly more applied, so doing mathematical modelling of infectious diseases. But again, I still don't know if I'm going to carry on in this same trajectory or change up. So it's really a case of just doing the things that I'm enjoying the most at the time. But a little bit about the modelling I did before COVID. So as I mentioned, I did an undergraduate degree in mathematics, and it was a 4-year integrated master's. At the end of that, I wasn't sure what to do next. But I saw a PhD advert that I thought looked really interesting, and it was on network-based modelling, which I hadn't done any of before. So I really just followed my interests, and you know, I decided like that looks really interesting, so I'll just go with it. So I did my PhD on network-based models at the University of Sussex alongside James and Edward. And yeah, I looked at models in a couple of different fields really, which was quite that's kind of another reflection really, is that I think it's quite useful to explore different research areas, especially if you're not, you know, entirely sure where you want to go. But very briefly, I did some work at the beginning of my PhD, which was looking at a toy model of a brain, effectively, a very, very small brain, and trying to look at how the spatial arrangement of different neurons in the brain would influence the kind of overall behavior of the brain. And then secondly, I worked on some projects which were more about networks describing people and the connections that we make with each other, and then looking at how disease spreads across those networks. And then after finishing my PhD, again, I wasn't really sure what to do even then, and I was considering leaving academia and going into teaching, but then I saw a research fellow position in the next department over, in the life sciences department, which looked really interesting again. So I took up that position. The research was actually really difficult, and I felt like I wasn't really making much progress with that. But on the positives, I took lots of opportunities during that time to speak and do public engagement where I could. And I found that this was something that I really enjoyed, kind of the softer skills of research and thinking about like storytelling. And I think it made me focus on what are the things that give me energy. And I think it's really important to try and recognize like kind of what feeds you and what drains you. And another reflection from that postdoc was attending conferences. So I was really lucky to attend a conference in America, followed by a workshop in India, all in the space of December of 2019. And obviously then I didn't know what was coming. And I found that it was just really good to go and experience these kinds of events and travel and network. And search engines are really great for finding these kinds of opportunities and developing links with researchers in other locations. And the other thing that I wanted to note was that... Is that a question? No. The other thing I wanted to note was that as an early career researcher, there's a lot of funding available to do these kinds of things during that stage of your career. So definitely, I would say to make the most of those funding opportunities and just keep doing like random Google searches. Because for example, the trip to India, I just happened upon this opportunity like on a random Google search one day, and I was like, oh, this sounds perfect. So I'd definitely recommend that. Then COVID hit. So during the first lockdown, as I mentioned, the research wasn't going very well, and my funding on that project was, I had about 6 months left. So I was starting to think that I want to change up here. So at that point, I applied for a few different jobs, and again, chose the one that I thought was most exciting, which was a postdoc at the London School of Hygiene and Tropical Medicine, working on the COVID response. So I knew it was something that was really important, and I felt like I could contribute here. And again, my reflections on this stage was that it was really good to apply for more than one job. And I noticed how I kind of had front runners at different stages. So at one point I was really excited to move to Paris, but then I ended up being really excited and thinking like, this London school job is the one that I want. So of course I started the job, a few months into the pandemic, completely remotely. And then I had to learn the ropes. So the context of me obviously arriving into this job, into this amazing team of researchers, some of whom I've got pictures of here. But yeah, it's quite scary to join, such a fantastic group of people and try to get up to speed so quickly. But yeah, I guess that was what I had to do. So I was trying to understand how this big transmission model was working, that was being used to describe community transmission of COVID in the UK. So I'm going to go into a bit of detail about the actual model and the research that we were doing. So we call it COVID-M. This is the model name, but it's a deterministic compartmental model. And as I said, it describes the dynamics of SARS-CoV-2 transmission. So in the model, we have age structuring, we have geographical structure. So we look at the different NHS regions within England. And then it follows a fairly traditional compartmental structure. So the SEIR there stands for susceptible, exposed, infected, and recovered. And the model also uses Google mobility data to inform contact rates. So as I said, I was kind of joining this big team when they're already doing great work, trying to learn a new model, and really having to kind of have faith in my own abilities and just like persevere, where it's quite difficult to integrate into a team remotely that is also, obviously everyone was quite tired at this stage. But one of the projects that I was working on kind of in the first few months of this job was, we in England, you might remember, we had some kind of tiered restrictions where different regions, like different, I think it was different local authorities, would be put into different tiers depending on like how hot the epidemic was in that location. So we worked on a study that looked at how effective those different restrictions were, and also compared the lockdowns that were going on in Northern Ireland and Wales. So we could kind of measure the effect of those lockdowns by using mobility data and looking at how much that had changed in response to a lockdown. And so we were able to look at counterfactual scenarios about, how if we implemented this style lockdown, how many deaths would we expect to see over a certain timescale. So yeah, this was kind of the first big project I was working on. And obviously, as with all of this work, it's a big collaboration. So it's not just my own work here, but yeah, many other names that were involved too. And then really not very long after that project was kind of getting published, we had the alpha B117 variant of COVID, which was first detected in November from a sample in September 2020. And at the same time, the vaccination rollout had begun in early December, I should say 2020. So one of the things that my colleague Nick Davies did very rapidly at that time was extend this compartmental model to allow us to consider two separate co-circulating variants of COVID. And I think my reflection from this time was, although it felt like I couldn't do a lot, like there were elements where I could contribute. So I think that it's really important to assess like what you are really good at and try to contribute there. So particularly something that was really good with this particular project, I mean, we got a lot of press coverage and we ended up publishing that in Science, which was really exciting. But yeah, a great thing that I was able to do was go on BBC Radio 4 and be part of their More or Less radio show. And even just getting that experience of talking about the research and what we were doing and understanding how a radio show is made, you know, that was something that I could contribute to. And then this section is a bit more about some of the modelling that I've been doing for SPI-M. So as James mentioned, SPI-M and SAGE are kind of these committees that provide evidence and modelling to inform government decisions. So the M in SPI-M stands for modelling, and there are also lots of different groups. So I've kind of come up with a schematic here. So you have like the UK government, SAGE, which is kind of directly advising the government, and then SPI-M, the modelling group, but you also have SPI-B, which is a group on behavioural insights. You have the social care working group and many others. So my work is really just a part of many different evidence generation groups. But what we do on SPI-M is we had like regular meetings throughout the pandemic, and we would collate real-time modelling and analysis submitted by various groups of academics around the country. And we would produce like various products. So whether that would be, there's a lot of groups that produce estimates of the reproduction number or the R number, and that would be like published regularly. And then sometimes there would be like ad hoc commissions looking at specific policy events or changes. So it really depended a lot on kind of what was going on with the epidemic, what we were having to do in SPI-M. But the example that I'm going to talk about here is some of the scenarios, modelling scenarios I did, I guess it would have been in autumn of 2021. So looking over the autumn and winter, you know, what do we expect to happen in that time? So yeah, they were asking for scenarios from October 21 until September 2022. And I thought I would set out here a bit the model we have. So I mentioned before we'd extended the model to have two variants, but by this point I'd extended the model to have a third variant, so that we could capture kind of the first earlier variants and the takeover of the alpha B117 variant. And then in the summer of 2021, we had the Delta variant. So we were able to capture the kind of emergence and spread of each of these variants over time using this 3-variant structure. And I'd also extended the model to have quite a complicated structure to account for vaccinations. Now, obviously, in England, at least, we were using a couple of quite different vaccine products. So the VAs and the VBs represent different vaccine products. So this was like the AstraZeneca viral vector vaccines, and then the mRNA vaccines. So you probably know the Pfizer and Moderna vaccines. Originally, we just had like 1 and 2 for each of the vaccines to represent the different doses. And this would enable us to allow individuals to be moved into those compartments and have an extra level of protection against becoming infected and having you know, more serious outcomes. But by this point, we were starting, I think the booster rollout began in September of 2021. So we added a third vaccine compartment to enable us to model that extra protection. And then, so as I mentioned, there's a lot of transitions here, so all of the arrows are not shown, but effectively from any of these compartments on the left, you could be infected by any of the 3 SARS-CoV-2 variants on the right. And then once you transition through the infectious process, you can have the S here stands for subclinical. So we kind of split people that are becoming infected into either subclinical or clinical pathways. This is preclinical and this is clinical. But eventually you will end up at the other side of this in the recovered compartments, at which point you can have immunity from against being reinfected, but you can eventually go kind of back round to the start. So you can lose your immunity and go back to the start, or be cross-infected, which means you would have immunity to one variant but not another. So it's quite a complicated model structure, let's just say that. And in terms of the ask that we get from SPIAM, I wanted to talk about the kind of timescales that we're doing this work under. So there's a lot of time pressure and really limited resources, both in terms of like computational resources, but also in terms of like human resource. So I think I've learned this the hard way. You have to prioritize your needs first. You have to learn to say no. Otherwise, yeah, you just the requests would be endless. But in terms of this specific project, I think the initial ask was in late September. But effectively, the real work that we were doing was getting the final specification from SPIAM on Friday, the 8th of October, and then delivering it on the Tuesday. So really just leaves kind of like a weekend to do all of the work. So we were estimating epidemic trends for England. Originally, they wanted it until March 2022, but they later extended that to September. They wanted information on what we expected infections, hospital admissions, hospital occupancy, and deaths to do. They wanted us to consider different behavioural changes. They wanted us to consider the effects of booster vaccines being rolled out. We had to consider the vaccine rollout for 12 to 15-year-old children. And then we had to make sure we were using the best available estimates and including sensitivity analyses for vaccine effectiveness, waning immunity, and seasonality. And then if we had time, in addition to that, they wanted us to consider what would happen if a new variant of COVID came along and it could evade the existing immunity that was in the population. And they wanted us to look at the effect of Plan B measures, which were specific non-pharmaceutical interventions, public health measures that the government had outlined that they could implement. So in terms of like what we actually do with the model, so we fit the model to multiple different data streams from England. And what you can see here left to right is the different NHS England regions, and then top to bottom are the different data streams. So we have like deaths, hospital admissions, occupancy, ICU bed occupancy. We use PCR prevalence data and seroprevalence data. And we fit the model to all of these different data streams. And then we also fit the model to the emergence and spread of the different variants of concern that I mentioned before. So first of all, this is the alpha variant. And then secondly, this was the delta variant. And then given a fitted model that has the best parameters given all of that data, we can then project forwards what we think is likely to happen under a number of different conditions. So in terms of the behavior, the main way that our model captures behavior is with the use of mobility data. Sorry, I've got distracted by the Formula One. But what you can see here is I'm plotting mobility data over time. And the way that this comes is they split it into different kind of metrics. So it's describing visits to different types of locations. So you can see like workplace, transit stations, so train and bus stations, retail and recreation, and grocery and pharmacy. And in grey, what you can see is the smoothed data for England throughout the pandemic. you can see kind of like a big drop at the beginning, and then drops at various different times due to lockdowns or Christmas and various different things, school terms. So we use the actual data as it was in the past, and then we make assumptions about what we think might happen to that data in the future. So we would look at different scenarios. So what if nothing changes? What if it really quickly increases to 1, which is the kind of average baseline level, which was measured early on before the pandemic. And then we could project forward based on these different scenarios. So we can look at different behavioral assumptions and how that would change the projection of the epidemic. So for example, here you can see having no change would project it in a large wave over the winter, but low levels during 2022 with a resurgence later. But for example, if mobility returned to baseline within a period of 6 months, then we could see like a much earlier resurgence in 2022 being projected. And then we could also look at other uncertainties. So it's quite difficult to measure how quickly vaccine protection wanes over time, so how quickly it reduces. And so we were able to implement scenarios with different rates of that waning. And we found that this is quite an influential factor in terms of when we're projecting forwards. And also something to note here is the dotted lines show like an individual epidemic trajectory. So you can see like sometimes these will fall out of the kind of average. So the kind of solid line is the mean, and then we have like quantiles here. And then we could also look at different scenarios for the booster vaccination rollout. So looking at, because at that point, I think boosters were being recommended for everyone over 50, but nobody under 50. So we were assuming that over 50s would reach 90% uptake relative to those who'd got their second vaccine. And then we could look at different scenarios for those younger age groups. And obviously, well, I mean, unsurprisingly, having much higher uptake of booster vaccines in a wider range of the population led to lower levels of transmission in the projection time period. So effectively, we wrap up all of this work, we present it into SPI-M. SPI-M will write consensus statements which get published, and then eventually these papers do also get published. So it's all kind of accessible to the public, and they can see what evidence we were presenting at the time. And then sometimes, depending on what we're saying, the work does get some media attention. So this particular one was, I think the point we were projecting was that cases would slump in winter. So this was some of the coverage that these particular scenarios got in the media. And then kind of contrasting that work, obviously you all know now we got the Omicron variant not long after that. So this was a bit of a traumatic period, but we had to very quickly respond to the Omicron variant. And I think we were very lucky at that point that South Africa had made the world aware of the epi situation and the Omicron variant very quickly. So that really enabled a lot of scientists to assess what the consequences might be for their individual countries. But when we were doing this work, I think Omicron was designated or first reported to the World Health Organization, I think it was the 30th of November. We managed to publish something by the 14th of December. So we really managed to do a lot of work in a very short space of time. But we had to kind of go from first principles, really. So understand, like referring to the genetic sequence of Omicron and looking at the mutations that Omicron possesses, and then comparing that to the number of mutations that previous variants had, we could really early on see that Omicron has a very large number of mutations, and given the rate that cases and hospitalizations were rising in South Africa, it was definitely really concerning. And what I'm plotting here is, well, this is from Mood, but it's showing S gene target failure data, which is one of the ways that we could detect Omicron. And we can see this very early uptick towards the end of November, which suggested that we were already, that Omicron was already potentially spreading in England. But one of the things that we really didn't know, like we kind of knew the epi situation, we knew something was going on, but we didn't have a lot of information about what it was that was giving Omicron this advantage. We didn't know if it was just inherently more transmissible, which the Alpha and Delta variants had been. But you don't really get definitive information on this for a little while until lab studies have been carefully done. So we're kind of going a bit on the knowledge about the mutations of that Omicron has, and guessing that we think that it's somewhat immune evasive. And very early lab studies, the first, I don't know, 3 or 4 came out within a day. But they were definitely showing that Omicron, had immune evading properties. And what we're showing here is the D614G is a COVID variant that was circulating in the summer of 2020, so kind of early days. And this is comparing to the Omicron variant. And what the studies are measuring is the level of neutralization in people's blood against these variants, or this sera against the variants. This is not my field, so forgive me if I get some of this wrong, but effectively what they're measuring is a big drop in the ability of people of neutralization against the Omicron variant compared to the pre-existing variant. So it's obviously looking quite concerning at this point. And as I mentioned, a few different studies, helpful in terms of thinking about how a change in neutralization might relate to a change in the level of immunity that we have. As I mentioned, we kind of referred to previous variants and looked at the number of mutations that they'd have and the measured reduction in neutralization relative to the kind of wild type original variant. And Omicron, by a long way has the largest number of mutations. So we were thinking it could be quite a significant reduction in neutralization and thus quite immune evasive. So we came up with scenarios effectively. Scenarios assuming different drops in neutralization. And this really excellent paper by Curie et al., what they had done, and we'd really relied on this quite a lot, is they had measured the neutralization level for lots of different vaccines and for convalescent sera, so people who'd been infected and recovered from COVID, and measuring the kind of neutralization level and relating that to your level of efficacy. And that's kind of, vaccine efficacy and effectiveness are sort of the same thing. Efficacy is usually measured in a clinical trial, whereas effectiveness usually refers to real world effectiveness. But this relationship here kind of allows us to, given one level, we can assume a drop and come up with another level. So then we could come up with different scenarios and assume different drops in protection with Omicron and explore how that would affect the projected dynamics. So I think I'll probably skip over this a little bit, but another really useful part of this paper is one of their modeled relationships between efficacy against any infection and then severe infection, and that enabled us to kind of scale up protection against different endpoints. So we can scale up protection against infection to more severe outcomes that we model. So for example, hospital admissions and deaths. And we were able to explore these different scenarios with our different assumptions and model kind of the takeover of this Omicron variant and then what consequences that might have. Again, we fit the model same as before to different regions in England and to different data streams. And then we're able to look at what we're plotting here in red is the proportion of positive cases in pillar 2, which have S gene target failure. So we could use that as a proxy for how many infections are attributable to Omicron. And we could kind of match up our different scenarios to the very early signal of the change in the S gene data. And then we were able to explore different scenarios. So doing nothing. So this is boost high, escape low. So we had that. This is just showing for that particular scenario, but we could look at what if we do nothing? What if we have plan B? What if we go back to previous levels of mobility, and what effect that might have on the pandemic? So yeah, we were able to show that we might need restrictions in order to suppress transmission to levels below the peaks that we'd seen before throughout the pandemic. And so this led to a lot of press, and it ended up, I think we were the top BBC News story, which was kind of bizarre. We had a press conference to, explain the work to media representatives. And I then started getting abusive emails, which was quite entertaining. And my boss very nicely wrote a nice article about what it's like to receive this kind of abuse. But I think my reflection on this point is definitely lean on your colleagues, friends and family, especially in this kind of high stress situation. And then finally, I want to talk a bit about what I've been doing in the last few months and kind of the future of COVID. So things have kind of calmed down since that work. Very luckily, Omicron turned out to be less pathogenic, so kind of less intrinsically severe than previous variants. And so our projections turned out to be on the pessimistic side because of that. So although we've seen extremely high levels of infection in the last few months, it's, we've kind of escaped without needing to go into very severe restrictions like a lockdown. And so since then, I've really been working on trying to consolidate the projects that I've been working on, and submitting a first author paper, which, as you'll probably know, if you're working in academia, it's one of the things that you have to do. And yeah, one of the frustrations I think is that working for SPIEM and producing these public health reports means it's really difficult to prioritize the metrics that academia values. So, submitting first author papers. So I think one of the reflections from the last few months is to focus on the small wins. And one of my small wins is coming up with like a new way to visualize the model. And spending a few hours in a Google Slides, coming up with all these transitions, but at the end of it being really proud. And so this was definitely a small win for me. But it got to the end of March and I had the paper ready to submit, and I was really happy with it. But then the Omicron BA2 sublineage was taking off, and we decided, okay, we need a rewrite of the paper. So I think my reflection on this point is basically don't give up. If you really want something, don't give up. But the moral of the story is I did get there in the end, and I managed to submit this paper yesterday. And so yeah, I've got this model which is fitted to data throughout the pandemic so far, and again, very similar story, projecting forwards into the future, what are the key uncertainties, what should we be looking out for, and what might come to pass. Some of the other reflections I had is, because I found these 5 months really difficult, probably more difficult than the high pressure parts. Recognize what you have done, talk to friends and colleagues, and also keep doing other things. So don't focus too much on the main worry. If you're getting energy out of doing teaching, then definitely do that, and buy yourself flowers. And finally, celebrate. That's the other thing that I wanted to reflect on, is that it's really important to stop and take stock and celebrate even the small wins. So yeah, as I said yesterday, I submitted a paper. It's online now, so you can scan the QR code and get to it from there. But in terms of all of this, all of this modeling and all of this stress, like, I wanted to think about what the reflections are, and this is kind of summarizing some of the key ones. So I think whether you're in mathematics or not, whether you're early career or not, I think it's really important to follow your interests and identify what excites you and what you're passionate about. And if you follow that, that will really make you stand out. As I mentioned, explore different research areas and activities. I think variety is key, especially in the early days. I think it's really nice to get experience in different fields if you can. Similar to what are you passionate about, what gives you energy and what takes it away. I think it's really important to identify those sorts of things so that you can learn what to say yes to and what to say no to. Particularly when you're early in your career, I think keep searching for opportunities to travel and attend events in person. Obviously, it's been quite difficult during COVID, but there is a lot of funding to take courses and attend conferences and workshops available, especially during your early in your career. So definitely take those opportunities where they exist. Keep multiple options open. Sometimes, even if you're not 100% sure on applying for a job or working on a project, it can be useful to go for it anyway, and maybe along the line you'll decide, no, this isn't for me, but maybe you'll change your mind and it will end up being something you really want to do. So I think always good to have multiple paths open to you. Think about how best you can contribute. What are your skills and attributes? So one thing I've definitely learned is that people have very different expertise, and so one person will be really skilled at one particular thing and the next will be skilled at another. So I think it's good to assess what you are good at, and maybe what you're not so good at, so you can maybe strengthen that. When there's time pressure, it's really important to keep balance between work and life and prioritize your own needs. Second to last, humans are very bad at estimation, and I've learned this a lot over the last few months. Things usually take longer than you plan for. And finally, be kind to yourself and remember to pause and celebrate. And before I finish, I have to acknowledge all of the different people that have contributed, because there's so many. So in particular, a lot of the work that I presented here is not just mine. Really, Nick Davies has done a huge amount of work, both before I even started this job and during. So Nick Davies has done so much, as well as John Edmonds and Mark Jett, who are my bosses. Carl Pearson as well contributed a lot in terms of the Omicron work that I've been doing. And then members in the Centre for Mathematical Modelling of Infectious Diseases. I'm really lucky to work in a group that has so many people and such vibrant conversations going on. So it's definitely useful to be in a large group of people who are interested in the same things. And finally, my funding. So obviously I wouldn't be doing it without my funding. So shout out European Commission and the EPIPOS project. And then also my friends, my family, my therapist, and my cat, who definitely I wouldn't have been able to do it without them. So I think that's the end, and thank you so much for your attention.
Thanks a lot. Yeah, I think the having prioritized time for yourself and those reflections, I can certainly attest to as well. That I really want to emphasize how incredibly stressful modeling COVID probably was. I think there's a lot of, especially with regards to the media, and I think John probably said this in his article, but that the time pressure and the stress is not really documented whenever you read the outputs, and that these are people behind screens who are trying as hard as they can to get these outputs that are asked for them by people who possibly don't understand what is required to put those together, and the interpretation is not always clear. So yeah,
that's a really good point. And one thing, I can't remember if it was some kind of like scientific technology committee in the government or something, and they were kind of like assessing the work we'd done with Omicron and asking like, why did we get it so wrong? And like, well, not that we got it very, very wrong, but we did the best with the evidence we had at the time. But one of the comments they were making there was like, oh, well, couldn't they just rerun the model like a week later? And I was like, the actual human element, like, yes, in theory, you can rerun the model again. In theory, you can keep updating it. But yeah, the human side is really not seen. And particularly, like, there was a limit to how much we could do. And the computational power and like what was actually possible, both computationally speaking and like humanely.
Yeah.
So I think that aspect has been quite hard because we've got a bit of slack. Yeah, we just get a bit of slack. People will say things on Twitter like, oh, you should go to prison. And it's like, I don't think they really see that we are generally just trying to do the best. Have good intentions.
Yeah, I mean, I had something to say now, I've forgotten. Oh no, it's gone.
What did you say? You're shamelessly going to steal the images?
Yeah, those images of the compartmental model are so good. Yeah,
Thank
they're
you.
so good. I was like, me and Edward have been trying to figure out how we can show multiple layers of compartments, and yeah, that was
honestly, you've made my day.
Yeah,
I was thinking that was super trivial, but
no.
I think it's one of those things again, like I realized I'm actually a very visual person, and I really enjoy that aspect of research. And it's funny, like, yeah, how much joy I got out of that, and even knowing if it's useful to you, that's good too.
Yeah, I said I'll cite it appropriately as well.
Yeah, if you go to my preprint, I have I think no way. Have I linked to this? I think I've linked to the Google Slides, or I can send you the link anyway.
Yeah. Does anyone have any questions before I ramble on about how difficult it was during COVID? Because I always have some, and I have two. So maybe I'll ask one if no one else has any questions. I want to know more about the Google mobility data, but maybe if I ask you that later, because I don't think it's that important. But I think the one which is probably going to be quite important to people here is, how did you manage the literature and the literature searching whilst developing the models? Because there was so much COVID literature. How did you manage to go through it all? Was there any technique that you used to sort of use some but didn't use others?
It's a good question. It's funny, writing this talk like made me realize how traumatized I've been. But I guess the simple answer is no. There's nothing like systematic. I mean, at times for certain papers where you were required to sort of do a systematic search, like we had to write scripts to pull all of the like COVID preprints and then search for like specific keywords within that and then like review them. Yeah, that's tedious. But in terms of a lot of the work I was doing, it was really difficult keeping up to date with the literature on vaccine effectiveness. Like, so difficult. And I don't even think it's possible. Like, I think that's one of the real challenges, is that so much evidence was being generated that it would be impossible for any individual to ever keep up. One of my colleagues, Nicola Lowe, who works in Switzerland, she's also involved in the EpiPose project. She had like a systematic living review of COVID literature, which they were like, adding to this. I guess the idea of a living review is you keep like looking at how much evidence there is. And obviously it's growing over time, but it got to a point where they couldn't actually do it anymore because there was so much. So I guess the answer to your question is, I didn't really do anything systematic. A little bit of it is like word of mouth within our kind of Slack channel. So people would be posting like, have you seen this? Have you seen this? But then a lot of the time, I think most of the time for me, it would be like, okay, well, I need to find a parameter for the rate of waning. And so then we would like have to search for that specific thing. I don't think it's possible to like keep up to date with like everything that gets put out there on every topic. So yeah, that's maybe somebody can come up with something better to kind of categorize and store the state of knowledge. But
yeah, I know someone at Imperial, I think, was keeping like a database of parameter values, I think, for like mathematical modeling. But I think this raises a very good point that there was, I mean, this is particularly in COVID. I can't say very much for other fields, but the mathematical modeling of COVID was outrageous amounts of stuff. I mean, we had the RAMP organization going through a lot of the literature and working out like what was usable and what wasn't. But yeah, I mean, there was a lot. So
yeah, the other thought I have on that as well is which would often make me a bit uncomfortable. It's like I'd done all this searching, and it wasn't entirely systematic. You know, if you were doing a sort of systematic literature review, you would do a lot more than I did. But obviously I'd come up with these parameters to feed into the model on like how good vaccines are, how well they protect you against infection and disease and hospitalization and so on. And my colleagues would like use the same values. But it always made me really uncomfortable because I'm like, well, what what if I'm wrong? Like,
yeah.
And I think that's another thing, like it's hard to assess, like if you're relying on like something that somebody else did, it's obviously easier to do that, but it's like, I don't know, it's really difficult. I just wish there was a way of like, yeah, classifying and storing knowledge, but
you can somehow like follow the literature chain of like where it gets cited, but then sometimes you end up with like a circle site and you're like, where did this value come from?
Yeah, and even actually, yeah, there's been a lot of times where you're like, a paper quotes a value, but then you have to do a lot of digging in the supplementary and be like, did they do this? Did they do this?
Yeah.
Did they do this? I hope in my papers I'd like explain things well enough so people don't have to do that, but
it's possibly going to be helped with the notion of like open access science now and like reproducibility. So like part of the reproducibility is, if you write an algorithm, can someone else develop it? And I think the same thing, I hope the same thing will happen with like parameter values, making it very clear what is in the literature and what is like scenario based will hopefully sort of like move forwards.
I have a question.
For yourself?
No.
Oh.
For anyone,
Oh.
which is what happened with the Formula One. Someone put it in the chat. I think maybe Leclerc qualified first, but I don't know. All I can see is Ferrari flags.
You can
I hope Leclerc's come first. He's in my F1 fantasy.
you can iPlayer it.
I'll look it up and I'll post it in the chat.
Does anyone else have any questions? No, I'm going to take that as a no. I will ask my Google Mobility data if you're still available later.
Yes, but I have to go at 5.
Okay, maybe I'll put it in the chat at some point instead. Okay, so let's move on to the final talk of the day. And again, I'm very sorry that we've run really far, almost 45 minutes late. We have Jane White from the University of Bath. She's a professor of mathematics working in applied maths. Her recent prominent research is working with colleagues in sub-Saharan Africa, using mathematical models in the context of criminal activity and crime control. She has also worked on models for the monitoring and delivery of drugs across the skin and collaborates with pharmaceutical companies. Jane has been awarded grants from the Leverhulme Trust, from the Leo Foundation, who I've never heard of before, but would love to hear a bit more about it because they sound very good, and EPSRC. She's held many prominent positions and activities in the LMS and is one of the founding directors of MASH, which I believe is only at the University of Bath, but I'm not absolutely certain, but is an institutional mathematics resource which offers a range of activities to support the mathematical and statistical needs for all members of the university. Welcome, Jane, and feel free to take over from that.
Okay, yeah, thank you very much, James. Let me just get going with this. Okay, So I think you can see my screen here. Yeah, so thanks very much for inviting me here. I hear what you say, Rosanna. I need to go by 5 as well, so I will endeavour to give a fairly concise talk. And actually, it's interesting being the last talk this afternoon, because much of what Rachel and Rosanna have talked about in terms of their career is probably stuff that I've already, things that I was sort of broadly going to say. I think there's some slight additions that maybe I can make in terms of the scope of an academic career, but in terms of the advice, I think you've already been very wisely spoken to this afternoon. So, yes, I, unlike the previous 3 speakers, have been at the university as an academic for actually 25 years now. So I've got quite a long, I've moved quite a long way from being an early career mathematician, but what I'm hoping to do is to kind of give some sense of what that academic careers look like, and relative to things that Rosanna, for example, was talking about making choices along the way. Okay, so the first thing in order to do that was to kind of think about what an academic career sort of generically looks like, for want of a better description. And these three pillars, I think, are typically quite commonly found if you look at career progression documents across the UK HE environment. If you're lucky enough to work at somewhere like Manchester University, there would be an additional one which is all around social enterprise and social responsibility. I'm not sure how widely that additional pillar has been added to at different institutions. It's not currently available in Bath, but there are these 3 core areas, and we've heard a lot today about research. We've heard a little bit about teaching, and we've heard a little bit about leadership and management. And I suppose for me, before I became an academic, before I did my PhD, I actually trained as a secondary school teacher and taught in schools in the state sector in the UK for a small number of years. I was there for 3 years. And so I think in my heart, teaching is never far away from what I'm thinking about, and education in a very broad sense. And so I probably would position myself, and always have, as someone who enjoys doing research. I found the talks that we've had this afternoon absolutely fascinating, and you know, want to learn more about the things that people have talked about. But if there was something that I would never be able to lose in terms of my career, it would be on the much more towards the teaching and learning side of things. So what I thought I would do is just talk through sort of some examples of what I've done during my career, and I've done it in a way that kind of mimics, if you like, the sort of process I've gone through. Not exactly, because the examples aren't in quite the same order as they would have been chronologically, but this idea that there is some research I'm doing, there's some teaching activity, some leadership activity, and it all comes into this pot, which is my career, and things happen at various times, and I prioritize things at different times. And I think one of the things that's particularly interesting about that in terms of an academic career is that for a career where you are you're employed based around your sort of research interests, that tends to be the focus for many people, and that is indeed, I was employed as a lecturer at the University of Bath, not because of my teaching, but because of my research interests. But there are also, of course, now pathways in most, if not all, universities where you could go in through a teaching route and progress, have a full academic career in that context. That isn't the scenario that I followed. Okay, so I'm now just going to do a series of examples to give you a flavour of kind of how I've sort of made use of these different elements through my career. So the first is an example of control of infectious diseases. This was done probably nearly 10 years ago now, together with the School of Social Medicine at Bristol, and it was around the introduction of a national chlamydia screening programme. And what we wanted to do was to build a model to look at control strategies. The complication for the control strategy was that in order to actually be able to describe something and understand the impact of particular components of the process, we wanted to take what was a highly structured network system, which is the social network in which people operate, and we wanted to move that from a network into a differential equation system. So just to give you an example, actually this isn't particularly a good example of a social network that would link to chlamydia, but it gives you the idea. You have these nodes and you have links to a certain number between each node, and you can get the transmission along there. So when we think about that in terms of an infectious disease model, we've got a very simple structure here where I is an infected population. We're thinking about the change of that over time. If we've got a population that is only separated according to susceptible and infected, and if we assume the population is constant, then we can create a differential equation that simply describes the infected population. But we have some terms from this in this equation that are directly linked to the structure of the network. And in particular, what we have is the incidence, so the interaction between susceptibles, which are the n-I, and the infecteds. So that depends on how the network is structured, whether it's a lattice, whether it's highly skewed, for example. And then if we are interested in a screening program, which is actually what we were talking about in terms of control, we have two elements of the screening. The first is opportunistic screening, and that is that people see the advert, they get persuaded to go and get screened, and if they're positive, then they get picked up through that screening. And actually, sorry, I forgot to colour this, but this parameter here, gamma, is a screening, so the opportunistic screening rate. And so that will pick up individuals who are infected, but as a result of that, by doing contact tracing, which actually no longer needs any explanation post-COVID, what you can do is you can pick up additional individuals who you can find because they are contacts of an infected contact. Okay, so this function here describes this tracing process, and the function f again will be determined by the shape of the network we're dealing with. So what we did is we simulated a network and we ran a whole series of infections on it to extract the shape of these functions. And what happened, you can see on hopefully on the bottom of this slide, is that they ended up being power laws. And this actually was quite reassuring, because in fact back in the 1980s there was work done by by an academic named Liu, who looked at very simple SIR models where the infection, the force of infection, had this sort of non-linear term rather than being a mass action, and described it exactly as this heterogeneous mixing of populations, which of course is what you get in a network. So there's some reassurance there, but what we were able to do then is we were able to look at our simulations and extract some information from it that fed back into our equations. So, for example, we looked at how the probability that a partner you would have would be infected as a function of the prevalence of infection in the entire population. And we could see differences in this depending on whether you were an infected individual, in which case you were more likely to have an infected partner, or a susceptible individual, and you were less likely to. So this data came from the simulations on the network, and it gave us some of the parameters in the previous equations I showed you. And then similarly, we looked at the connectivity. So K is the average number of partners, and this is the probability that you would have it, depending on what category of individual you were, whether you were infected, whether you were being screened, or it was in the entire population. So we took that information and we had this sort of semi-empirical then mathematical model, which was an ODE system that we could make use of to look at the different control systems. So with that, we then went ahead and looked at exactly that using sort of dynamic optimization. And what we were interested in then was the difference in different types of objective functional. So what were we trying to achieve? And actually, I think this was one of the things that was very interesting at the start of the COVID outbreak, because some of the decisions that appeared to be made that certainly you know, neighbours around me and friends who weren't sort of don't really have any sort of background in mathematical modelling were kind of not really appreciative of the fact that depending on what you're trying to achieve will give you a different protocol for your control intervention. So in the first case, we were going, okay, over a period of a year. So we were doing this over a period of a year. Let's suppose that the end point you are targeting a prevalence of 5% in your 5% in your population, and it's starting from a prevalence of around 8%, which was the data currently back in 2012 or whenever. So we were trying to get as close to this target as possible at the end point, capital T, but along the way, what we were also trying to do, the C1 and the CIs are all linked to the cost. So this first term is the cost of screening. This C1 is the cost of screening a susceptible, because there's no benefit to that. This is cost of screening infecteds, and then this is the costs associated with the contact tracing thereof. So we have these additional costs. Okay, and then we went about deciding how often you would want to update your controls. And so in what I'm showing here, we were assuming quarterly updates of control, because some of the work that had been done prior to this would allow continuous updating of controls, but of course that's a very difficult thing to do, because what the control is saying is it's saying what percentage, for example, of coverage of screening are you attempting to get in order to do as best you can here. And you can't change that on a daily basis. You probably can't necessarily change it on a quarterly basis, but the point was to try different timescales. And so we ended up with predictions that kind of gave you high levels of tracing required, relatively low levels of screening. But then if we looked at how our ODE compared with the underlying network, which obviously is the thing we're trying to mimic, we could see we got a fairly good fit for it. So going from the quarterly, we then went, okay, what if we update our controls monthly? And I suppose now we have seen that we do try and change things very rapidly, but probably not for things infections that have been in the population for a long time. And you can see that you end up getting a control strategy which is much more variable, on off, well, not exactly on off, but low high levels, changing quite regularly, probably not as achievable for many of these endemic infections, as I said. And what we were able also to do is to look at the costs. So those C1 through C4, they all had values that were estimated. And what we had is that if we changed our control strategy quarterly, then actually it did end up costing us more. This is the mean and the I guess the 95 interquartile range, and this is the changing monthly. And so you can bring your costs down, but of course we're not accounting at all here for the cost of actually changing people's roles within their particular jobs. And then just to give you an idea of other things that we looked at fixed budget. And so that's saying that actually we've only got so much money to do both our screening and our tracing. We've got a fixed budget, and over that we were attempting to minimise the final prevalence at the end point. And you get a different profile compared with what we saw before, a bit more on off for the quarterly controls. And similarly, if we have monthly controls, well, in that case, we have something looking quite different from the monthly, where we were targeting an endpoint prevalence. Okay, so that's an example of some work that I did probably about 10 years ago. And at the same time as I was doing that, I was setting up MASH. So as James said, MASH is the University of Bath Maths Resources Centre, but I mean, it's essentially a maths and stats support for all students and staff at the university. Rachel talked a little bit about something in Sussex around stats advisory for staff. Well, I guess MASH incorporates that as part of it. It's not the only MASH that I'm aware of in the UK. Sheffield also has one, but it is completely distinct from that. And I set up MASH actually, well, partly because I'd been working at support mechanisms for undergraduates in the maths department, and an opportunity came up to get funding for an institutional centre, and I took advantage of that, actually because for me at that time it felt like it was the right thing to do, because I wasn't in a position, or wasn't willing to be in a position, to travel very much, and I wanted to actually have something that was very tangible and that I could see evolving in a way that sometimes my research would take me a lot longer and I would find it harder to get motivated to do. And so MASH was excellent for me. I set it up, I got funding for that, and I had a whole series of grants from both institutionally and nationally to develop different areas of maths support, such that Bath is now nationally leading in maths accessibility, and all the work around things like Maths Jacks has been really driven forward by people coming out of the MASH Centre in Bath. Whilst I was doing that, people have talked a lot about networking during the day, and I would agree that that is hugely important. And so when I got funding for MASH, I also got funding to set up a regional network for colleagues also working in universities in maths support services, because at the time, back in, I guess that was about 2007, there weren't, these things were emerging. They're now pretty much everywhere, but at the time, staff who were working there could feel quite isolated. So we set up a regional hub, which then became this national network, the Sigma network, which is a professional body. And so we've now kind of got Bath is still involved in that. I'm not actually involved really at all in MASH anymore, but it was a hugely significant piece of work that I feel that I did at the university, because it suited me to have that type of role for a period of time. Coming back to research, another area I worked on, and this was collaborating with colleagues actually in the university, in the Department of Pharmacy and Pharmacology. I've done a number of pieces of work around drug monitoring and drug delivery across the skin barrier. So the thing about drugs, as I guess it's probably quite obvious to most people, is that usually you take a drug, so you might take it orally, and then it has to distribute in your body to the point where it needs to be, where it needs to actually have an effect. What you have to be very careful of is getting the drug dosage right, such that you are in the correct region. So you don't want your drug to be at too high a concentration to cause toxic effects, and you don't want it to be too low that there's no point in taking the drug, it's just adding chemicals to your body. And so it's really important to be able to monitor drugs, and in particular drugs such as lithium, which is used to treat bipolar disorder, where there's really actually quite a narrow window of therapeutic effects. So lithium is the first drug that I worked on, and I'm just sort of really going to talk around that a little bit, because one of the interesting things that was found is that when data was collected about fluxes, the amount of lithium you could extract from the skin surface, when it was in dead skin, so that's on this left here, over time, as you increase time, you extracted more and more. So this is cumulative amount here. Actually, no, sorry, it's not cumulative amount, it's amount in each of those intervals. But what happened when you did that for patients, so you extracted with someone who'd been taking lithium over a period of time, it was actually your first data point that was the largest, and then it decreased. And so what that was suggesting is that for some reason in that first half an hour you were getting far more lithium out of an in vivo subject, and that must be because the drug is available to be extracted, and that was what we started looking at. So we thought about how to describe this. This is a simplification of actually what's very complex, highly structured system. We have a stratum corneum, so that's the outer layer of the skin and the tissue. We have bound and unbound drug, and we have drug that moves in two ways. First, by diffusion. So actually it would take a tortuous route, because these cornea sites here, it has to move around, but we kind of use Occam's razor and think of the simplest possible. And also desquamation, and that's essentially the process by which we shed skin. And so what happens is we have our skin layers move up from the bottom where they with the epidermis, and eventually they shed off the top, and there's a velocity associated with that. So that's more like a convection type term. So with that, we had a very simple model. We assumed there was no lateral flow of the drugs, because there was no forcing. There was no reason for things to be forced in any direction, and also there's some empirical data for that. For the unbound drug, it would diffuse. It would also flow towards the surface because of desquamation, and it would become bound, and bound would become unbound. So we have this sort of flow between bound and unbound. And for the bound drug, that would simply move towards the surface because of the desquamation. Okay, so very simple model. And what we decided to do with that before we looked at the comparisons with the actual data was to think about the impact of the model parameters. And in particular, if we looked at the diffusion coefficient, the size of the reservoir, so what's actually in that stratum corneum in the outer layer, as the diffusion increased, we saw that from our very simplistic structure, we ended up with a more variable amount in the reservoir on a daily basis. And that's because of the interaction between the flow through the skin and the binding and the unbinding rates. And so if it flows through very quickly, then the rate at which things become unbound doesn't keep up with that, if you like, to then be the next amount moving through. And so you get this rate of oscillation going on. The opposite's true if D is small. And the reason we're interested in the diffusion coefficient is because it's linked to the physicochemical properties of the drugs through partitioning, so the affinity of the drug to be in lipid or in water, and its molecular weight. And so that's where we really link back into what they're interested in the pharma industry. We also were able to demonstrate non-compliance because in this case, you probably can see we've got an example where a patient is supposed to take a daily dose, but they only take every other day. And you can see this total reservoir size is significantly lower than what I had previously. Then if we want to compare the monitoring mechanisms, so how do we actually see what is coming out of what is in that outer layer? There's two approaches commonly used. The first is tape stripping, where essentially it's like you put a bit of sellotape on your surface of your skin, you extract it, you weigh it, and you calculate how much chemical is in it. And so you can only do that to a certain depth for protocol reasons. And so you only go to a certain depth D. And there you would pick up both the bound and the unbound drug. By comparison, reverse iontophoresis is a mechanism where you put in a small electric current, you apply a small electric current to the skin, and it opens up a flow, and the drug molecules can then move through with that flow. And what we do here is we separate that highly structured outer layer, the stratum corneum, into aqueous and lipid regions, and assume that it's only in the aqueous region where you would actually flow because of the electric current. And so what we're able to do then is to look at how the measurements you would get from those would differ. So for example, if the binding rate is larger than the unbinding, what you find, if I just look at this bottom one probably, is that you would pick up more drug measured through your tape stripping, because there would be more bound drug, and that would measure it, than if you did your electric current version. And by comparison, if the unbinding is larger than the binding rate, you would have more of your drug in the aqueous region, and it would get extracted more. And so one of the things you could do with this type of information is you could look at kind of in different scenarios, you could look at the likelihood of having binding versus non-binding in the skin for different drugs, because you could look at this comparison. Okay, so that then brings me into my academic, back into the teaching and learning environment, and just a couple of things here. The thing that I am probably most passionate about is assessment. I'm not so bothered about content, and which might sound slightly heretic to say, but it's true. What I think is important is how we think about assessing students and determining what they can do with their mathematics, what they understand by it, and how they think about it. And so I've been involved in a number of projects in the maths department, changing and modifying slowly, very slowly, assessment processes, speeded up a bit over the last couple of years, but in two directions. One is in terms of the depth. So if you go up in this vertical region, it's saying we're getting to more complex ideas, we're having to think more complicatedly. But in the horizontal, what we're going from is from entry point to graduation point, and going, actually, what should assessment look like over that period of time to see that we've developed these mathematical thinkers. And I would argue it probably could start with some knowledge, sheer knowledge testing, and simple techniques to test that people can do certain techniques. And then it should evolve, possibly through working collaboratively with other peers, to becoming someone who can talk about their mathematics and can answer questions about their mathematics. And so this is work that I feel, you know, is probably the primary thing I do in terms of teaching and learning, and was part of my my application when I became a National Teaching Fellow in a few years ago. Alongside that, nationally, the thing that I'm really interested in, probably because I was a teacher at one point, is closing the gap, or yeah, it's not really closing the gap, but it's completing the loop. For many years we have talked about the problem in this country and internationally about not having enough qualified mathematics teachers, and it seems to me that maths departments are exactly the place where we train the people who could become the suitably qualified maths teachers. Not all of them, but more of them than currently. And so I managed to persuade the LMS to give us a subcommittee of the Education Committee, and we're doing that exactly now. We're trying to find mechanisms, so you may hear about that if you're in a maths department anywhere, whereby we're asking maths departments to commit to encouraging students to think about maths teaching as a career, and really excited the fact that the Department for Education has now come on board with this, and so we hope to be moving this forward quite significantly in the next year. So then my final example is the work that I've been doing over the last 4 or 5 years in sub-Saharan Africa. And this is something that I really enjoy, because for me, it's the interface of teaching and research. It's working with PhD students, or mainly junior lecturers, to actually build up their research skills so that they can pass them onward in their teaching and learning. As a result of this, I'm currently a visiting professor at the University of Johannesburg, and this is how I know Edward, through some of the workshops that we've been involved in over the years. So just a very quick summary of one piece of work that we did. When we first started, one of our colleagues in South Africa said, I'd like to look at crime dynamics. That was it. They didn't really have any more focus than that. And so what we ended up doing is thinking about, okay, well, that sounds good. We need to think of a question. We need to think of a structure. And the thing that we came up with fairly quickly is that the data we have for crime is on number of crimes, but actually the process by which crime happens is through the criminals. And so we needed a linkage between crime and criminals. And we managed to get some data. This is the Western Cape, so it's just near Cape Town, and we were able to quantify it according to being areas of high crime, which was essentially, I think, more than 1 murder per 1,000 individuals, and green being low crime per annum. Okay. So as I said, we were interested in number of crimes number of criminals, but we had number of crimes. So we just kind of put them all together. We said that we have these crimes going on. A is in the baseline region, B is in the heightened region, and it depends on the number of criminals from the baseline region and the heightened region. And then we had the dynamics on the number of criminals, in a sense that there were opportunities for individuals to join the criminal activity. This may be opportunistic in two ways. One is that they might just kind of naturally, because of their environment, take part in some crime. They might see that crimes are happening and so they might join, and then there might be a peer pressure, this term here. And what we did with that model then is we tried to parameterize it, and we did that using very limited data over about 5 years. So the first 5 years here gives you number of crimes, and we're fitting our crime model to it, and the subsequent years are us looking at how good the model was. And in fact, what we saw ultimately is that we seem to be having a reduction somehow in criminals in the low crime regions, but we're heading towards this sort of steady state, which is a stable steady state. Okay. So then the final thing I wanted to say was about something that I feel has been growing in importance for me over the last 3 or 4 years, and that's around leadership and influence. I was elected as something called the Chair of Academic Assembly, which kind of supports the entire academic community in Bath, but that gave me an opportunity to be part of the University Council, and that was hugely interesting and rewarding because it gave you an oversight of the entire organisation. So not restricted within your own department, not within your own discipline, not within the purely academic side. It was the whole infrastructure. And during that, I was able to influence the decisions around declaring a climate emergency at the University of Bath, and then we've now got a climate action framework, which includes really quite challenging questions around how we would in the future decide to travel for meetings, something that you've been talking about earlier, and what's okay and what isn't. And alongside that, have started writing articles for these sort of more opinion or policy-based sort of organisations. So, for example, in One Key, what I did recently is take entry requirements for universities in maths departments and look at the percentage of students who actually achieve the entry requirements, that the offers that they're given, because one of the things we spend a lot of time talking about is that students really suffering because of the stress put under them, and it was clear to me for a number of years that one of the things we could do to alleviate that stress is have realistic entry requirements, at least at that point. There's subsequent things, but that's one of them. And it was really quite clear when I did the data, when I analysed the data in as much as you can get it, that so this is a bit weird to read, but essentially if you look here, probably just I'll look at this one, this is saying that about 45% of institutions, only 45% of institutions had between at least 3/4 of their students meeting the entry requirements over a 3-year period. That means that everyone else had less than that. And so my argument is that we should have a different mechanism by which we are allowed to decide what entry requirements we have. So finally, just to reflect on this navigation that I've had, I think for me, I've taken advantage of a sort of very broad academic career, research, teaching, and the leadership and management. And I've done that at different times in different ways over the last 25 years. Without question, the research allowed me to travel and allowed me to build up capacity in a number of ways. But also at the same time, the teaching bit of it was the bit that drove me forwards, and ultimately it was the bit that got me promoted. The leadership and management, I suppose, is the bit that has allowed me to start to influence things, and I suppose that's becoming more significant for me now as I feel I've got more things to say. And then just finally to point out, I very rarely show any pictures of my children, and I don't have any in my office, and I think that was intentional, although I don't know why necessarily, except that to say that, you know, for me that was a huge priority for many years. I was part-time for, well, nearly 20 years actually, while I had the kids, and I think one of the things that afforded me was that my career afforded me was the flexibility to do what I wanted, but it also meant I had to make choices about things that had to be realistic. I could and I couldn't do during that period of time. Okay, I'm going to stop there. Thank you very much.
Thanks a lot for the talk. It was really eye-opening in terms of different career paths. I think a lot of, I certainly speak for myself, but a lot of students and even going through PhD to postdoc don't really recognise the three different avenues of what a university does. A lot of outreach, I've been part of some outreach talks which actually discuss what does university do, and it's not just about teaching, it's not just about research, but all these other mechanisms and how that contributes to being a member of a university and working there. So I know that you are pressed for time, so do you have like time for one question, if there are any?
Yeah.
If the answer to that question is no, then that's absolutely fine.
No, that's fine. Yes, that was my fault for not being quick enough.
No, it's my fault for going over by half an hour. I don't know. I mean, it doesn't seem like anyone has any questions. No. So, like, I think that's. I mean, I have one. And I think it's going to be a quick one, which is just for perspective. Each of the projects that you mentioned, what sort of resources did you have available to you, and how long were they taking?
So, the HPV one, I had a PhD student and colleagues in social medicine in Bristol, but it was the PhD student was mainly doing the work. The drug one, I had PhD student, then we did have a research associate with one of the grants that was linked to that. And then the crime dynamics is kind of this consortium of people. So yeah, I try really hard to get the junior lecturers to do the work, but Edward was certainly involved in doing some of that data analysis. So I think for me, actually, people talk a lot about getting grants to do to get postdocs and stuff like that. What I did is so I've line managed lots of people, but they were in the context of MASH. So I got funding, but some of it was grant money to start with, and then it was getting those people permanent positions in the institution is how I kind of focused it. So it was slightly different.
That's very useful to know. Rosie, a quick one.
Yeah, thanks so much, Jane. I was just wondering about MASH in particular. Where did that funding come from? Was it like a scheme that is still going now, or like what, you know, I'm not really aware of schemes in that area.
Yeah. No, well, yes, it did come from funding. There was a national centre for excellence in teaching and math support that was based out of Loughborough and Coventry. It's still, I mean, well, those two things still exist, but the thing that doesn't anymore. So I don't think that funding is there anymore. I think the expectation for math support now is that it comes from institutionally, because it's just viewed as a standard offering. I mean, yeah, I've been here so long that of course it's quite weird talking about it, because it was very not standard, and certainly wasn't standard in somewhere like Bath, that was viewed as a sort of high profile university where all our students are very, you know, we get good students and stuff, but it's played a hugely important role. And yeah, it works a lot with the now with the access and participation plan, so supporting students to transition into university.
It sounds really amazing. It's not something I'd heard of before, but I'm gonna look it up.
Okay, good, please do. Yeah, they're a great, I mean, I can say that now because I mean, yeah, I'm not part of it now, but that they've been remarkable in what they've managed to achieve. So
thanks, Jane.
Okay, I think that's probably enough. I won't keep anyone else here for too much longer now. So thanks everyone for coming. Thank you all. I guess I hope this bit goes to YouTube, but thanks all of the people on YouTube who are listening to these talks. I must admit, me and Edward were just talking on the side, but this has probably been the at least I feel for the last few years, the best conference in terms of content that we've had. I'm really happy that we managed to be able to do this and that everyone sort of came. And I'd again like to thank all the speakers for today. I know most of them are probably left by now because we overran a bit, and so sorry about that. But yeah, thank you all for spending your time to come up with these really amazing talks and to sort of like discuss being what an early career mathematician is and how one progressed through that, and different projects that were available and collaborative opportunities. So with that, I'll finish. I'll hang around in the Zoom call for a while if anyone has any further questions for me about the IMA ECM or about anything else that we do in particular. But as far as that is, I think you're all free to go and enjoy the rest of your sunny Saturday.
I'd just also like to say thank you to all the presenters, and thank you to James for organising this event.